A very good morning to all of you. First and foremost, please let me know if the audio and video are clear. Good, wonderful. And many of you have wished Happy Teachers' Day. Thank you so much for that. I'm just waiting for 30 more seconds. I see that some of you are still joining, so just give me 30 more seconds and then we shall start. All right, so let's begin with the session. So yesterday we had begun with biotechnology and we discussed about some aspects of biotechnology, the basics of what genes are, chromosomes are, etc. And then we had begun branch by branch where we discussed about red biotechnology first, where we discussed about all the health applications related to biotechnology. And then we had moved on to agriculture and we also understood some of the basics of how biotechnology can be used in agriculture. Now, in this particular scenario, we have been talking about transgenic crops, right? We discussed that there is a crop, or let's say any crop, where you are bringing a gene from any other organism. You would say that it is a transgenic plant or a transgenic crop. So, today we are going to begin with the same topic. We are going to continue with the same topic. And that's why the first topic for us today is the development of transgenic plants. That how exactly do we develop the transgenic plants is what we are going to start with. So the basic technique that is used in making these transgenic plants is called as recombinant DNA technology. All right, it is called as recombinant DNA technology. So we'll try to understand what do we mean by this recombinant DNA technology and how does it help us in getting these transgenic plants. So recombinant DNA technology. So first and foremost, when we say that it's a recombinant DNA technology, the reason is that when we say this particular term that we have right here, recombinant DNA, recombinant DNA. So when we say recombinant means that there is a combination of two different kinds of genes, or let's say two kinds of DNA material together, and that's why it is being called as recombinant DNA. So let's look at this process and how exactly this process is executed. So first and foremost, what we do is we take the gene that is required. All right, we will try to isolate the gene that is required. This is the gene of interest for us. This is the gene that contains that one property that we needed to insert inside the plant. So suppose this is the gene that lets the plant talk. All right, so this is the gene for talking that, let's say, us humans have. So we take this gene from humans, and then this gene basically will be taken in its pure form. And once you have this gene, so this blue colored part, this is the gene of interest for you. So, for example, what we discussed about beta carotene in golden rice or BT in BT cotton. So, this is that particular gene that is of interest for us. Now, after choosing this gene, we also have to choose a vector. We have to choose a vector, or what we also call as a carrier. So, vector or carrier will be the carrier, meaning that this will carry the required gene. Now, this vector or this carrier could either be a bacteria or it could be a virus. All right, it could be a bacteria or a virus. In most of the cases, it is a virus, but it could be a bacteria as well. All right, so it could be a bacteria or it could be a virus. Now, what we do is in this bacteria or the virus, we find a relevant position where we can insert this gene. All right, we find a relevant position where the gene could be inserted. So if you see in this particular gene, this part, This is the where we are going to make the cut. And if you see, if you look closely, the cut is being made in a manner that you will cut it open, and then this gene of interest will be inserted here. All right. So you have to recognize this site. This site has to be recognized, and it's not that you can just make the cut anywhere. It's not that just that you have something, you have a genetic material, you just make a cut, you insert this, and you're done. So unfortunately, things are not that simple. That it's not simple that you will simply use this and let's say you have the gene of interest, you take the gene of interest, insert inside the plant, and you are done. I hope, I wish that the things were that simple, but unfortunately they are not. So first and foremost, what happens here is that we have to recognize this site in a vector. So this will be done with the help of certain enzymes which are called as restriction enzymes. All right, restriction enzymes. So first this area will be chosen, and then restriction enzymes will make the cut at the relevant area. All right, it will make the cut. Now it has made the cut, and it will open it up, and this is where the gene of interest will be inserted. So if you see, it looks something like this now, that you have made the cut. So now this is how it is. Correct, this is how it is. So you have made the cut from both the ends like this, right? So in this particular scenario, this is basically, let's say this is how now the vector or the carrier looks like. So in this relevant site, now what happens, the gene of interest is getting inserted here. So this is the gene of interest that we had chosen from this particular site, right? So this basically, this blue colored part, this basically comes here, and this is the site, this is the site where we have made the cut, right? So now that this gene of interest has come here, the next thing here is to make these ends to stick together. So you would need a fevistick, correct? You would need a fevistick or a fevicol to join these two parts. So this fevistick or the fevicol that we have here is called as the DNA ligase. This is called as DNA ligase. So DNA ligase is that fevicol, or it is an enzyme basically. It's that enzyme that helps you stick these two ends together. So now this DNA that you have, where you have the carrier and you have the gene of interest, this is called as the recombinant DNA. All right, this is called as the recombinant DNA. And once we have the recombinant DNA, this is where now it will be inserted inside the plant. But even before that, what we do is that first we take multiple photocopies of it. All right, we go to a Xerox shop and we ask for multiple copies, and by multiple I mean thousands of copies. All right, thousands of copies will be now made out of this recombinant DNA, and then it will be inserted inside the host plant. And then we'll hope that the host plant also recognizes it, can take this as a part of the structure of the plant, and then that plant also starts showing that property. All right. So in this manner, we see that this is how the things work out. This is how we look at the recombinant DNA technology. All right. So just to repeat what we are doing, first and foremost, we have to take the gene of interest, right? We have to find out the gene of interest, and we have to take this first. And once we have found the gene of interest, this gene of interest will go inside a vector or a carrier, which rather be which will be a virus most of the times, but some cases bacteria also. Now, in this, we have to recognize the site where the cut can be made. So we find that particular part. Now, just remember that it's not that easy to find this part. It's like a very big jigsaw puzzle in which you are trying to find one missing part, and you want to see where that missing part will set. All right. So to find that area, it's a big task. Otherwise, it's not that you simply will take this DNA of interest and you'll insert it anywhere. Doesn't work. You have to find that exact spot where you can do it. So now you have, once you have done it, this is how once you have made the cut with the help of restriction enzymes, this is where now you'll see that the DNA, that the gene of interest that you have, this basically will be inserted inside this particular host, and this will be made to stick together with the help of another enzyme called as DNA ligase. Once that has been done, This is where you create multiple copies of it, which is also called as polymerase chain reaction, or PCR. And then finally you insert it inside the required plant. So this is basically how the things work out in case of recombinant DNA technology. So first step is to isolate the desired DNA in its pure form. All right. Then after that, cutting the genes at the restriction recognition site. This is done with the help of restriction enzymes. So this is where they will determine the location, and after that the cut will be made. And after this, the step of ligation or joining the two pieces. So joining the two pieces will be done with the help of an enzyme called as DNA ligase. It will be called as DNA ligase. Once that has been done, we create multiple copies, which is called as polymerase chain reaction. All right. So single copy of DNA into thousands to millions of copies is created. And then finally, insertion of this recombinant DNA into the host. And this is where this process is termed as transformation. So transformation is nothing but inserting it inside the host. So in this process will be carried. Now, up there are some of the other applications of biotechnology in agriculture as well. For example, biofuels. We discussed about biofuels. We discussed that most of the biofuel still in the world is first generation biofuel, and first generation biofuel is based on what? It is based on food crops. So if you are using food crops for making a fuel, that will also can be called as biotechnology, because you are using a living or a recently living organism and you are processing it for some kind of application. So that's why biofuels will also be called as an agricultural application. Then after that, we have the concept of biofortification. Now, what we discussed with golden rice yesterday was that in golden rice, you are adding vitamin A. So this kind of a process is called as fortification. And when we are trying to do that with the help of biotechnology techniques, it will be called as biofortification. So fortification basically is addition of an ingredient to food to increase the concentration of a particular element. So there are various ways. Golden rice is an example of biofortification where we are using biotechnology. Some of the methods we don't use biotechnology. For example, salt, common salt, you will see that uh, it has added amounts of iodine. We call it as iodized salt, right? So this iodized salt also is fortification, but not biofortification. Biofortification is when you'll be using some kind of biotechnology related method for fortification process. So biofortification will be done to for the development of micronutrient dense staple crop. Now, micronutrient dense, what do we mean by micronutrients? Any idea? Yes, Deepika, you are correct. Anyone else who knows? Yes, Yashpal, you are correct. Starly, correct. Janvi, you are correct. Abhijit, correct. All right. So many of you get what we mean. Micronutrients basically are the nutrients which are required in very small quantities by the body. Required in small quantity. The example is vitamins and minerals. So these are called as micronutrients. On the other hand, you also have macronutrients. Macronutrients which the body needs in larger quantity. So required in large quantity, where you have the others. For example, the carbohydrates, fats, proteins. So these will be called as macronutrients because they are required in larger quantities. So in this particular scenario, so that's what I mean. If you go to a dietitian, if you, let's say, if you are overweight and you go to a dietitian, the dietitian will say that you have to control your macros. All right. So that don't eat a lot of carbs and don't eat a lot of macros. So these macros are nothing but the macronutrients, and carbs are nothing but the carbohydrates, right? So that basically is what we have to do. So that's why when we talk about fortification, we often see that fortification is not required for macronutrients because they are very easily available, and the body also craves for it. So body ensures that you get a lot of carbs, you get a lot of fats, etc. So it's that it's body's way of ensuring that body always has enough of energy to function. So that's why you'll always crave for more of carbohydrates, that you want to have more of chocolates and all that, and you don't want to have fruits and vegetables. And that's your body's way of saying that I need a lot of energy, so let me have every kind of junk food that you can, so that you give me all the carbohydrates that's possible. All right. So that's why fortification largely is required for micronutrients, with vitamins and minerals largely. So that is called as fortification. So in the food whenever in the food itself some kind of nutrient is added, we say that it is fortified food. On the other hand, we also have a concept of supplementation. So supplementation is when you take supplements. So for example, vitamin D tablets, vitamin B12 tablets. There are so many health supplements that are also available in the market. Protein supplements, many who are into regular workouts and uh, they would take protein supplements. So there are so many supplements which are also available. So when you take them directly, it is called as supplementation, and when you are adding nutrients to the food, then it is called as fortification. Now, in this particular scenario, there is one particular issue that has been flagged by the WHO, and last few years WHO has been talking about this concept of hidden hunger. Now, hidden hunger is a concept where we are saying that although you are eating enough, but you are not getting all the nutrients possible. Now, this is not because of choice. This is not because of somebody's bad food habits or bad eating habits. It's when most of the times in scenarios where food supply is being made. So, for example, let's say for a poor country somewhere in Africa or Asia, where let's say though that country is not capable enough of producing its own food and is completely reliant on some of the other countries to supply that food. Now, whenever these food supplies are made, you'll see that there will be a lot of grains, a lot of rice, a lot of wheat, etc. But not a lot of things that contain vitamins, not a lot of things that contain minerals. So, what is happening in these kind of cases is that although they are eating, although they are not starving, and you can't say that uh, they are uh, grappling from hunger, it's not that, but yet all the parts of the diet that is ideally required for the body to function and grow and develop, all those things are not really being given to them. So that's why in that particular scenario it is called as hidden hunger, that although you are eating, although they get their food, but they are not getting all the nutrients that are required for a healthy living. Okay. So hidden hunger, when enough food is available, but it lacks in certain nutrients, especially vitamins and minerals. All right. So this is basically what they call as hidden hunger. So if you see, there are a lot of crops where we have seen that some kind of biofortification has been carried out. So all these are some of the biofortified foods. So if you see, we have seen that in wheat we have tried to enhance the zinc content. In maize we see that higher level of vitamin A has been found. Sweet potato, we see that for beta carotene, basically for vitamin A. Then in case of in case of beans, we have seen that more iron and zinc content is added. Then we have the pearl millet, where we see that increased iron content is added. So in this manner, we have seen that vitamins and minerals are being added to all these basic types of biofortified food, and hence they all are being used in one way or the other, so that we can actually get an idea, or we can get the required nutrient. And especially remember that these are being done especially in areas where some kind of deficit, some kind of deficiency in people has been found. So those kind of areas, we often see that these kind of biofortified food will be utilized. Then after that, using them as animal feed also, we see that in many of the cases, we make the use of agriculture in making the crops, in developing the crops which will be healthier for the animals, which will have all the required nutrients for the animals so that they can function well. So, and also to ensure that with lower quantity of food we can provide all the nutrients that are required for the given animal or the livestock. So instead of, let's say, the kind of nutrient that they could have gained from a 20 kg consumption of food, can the same kind of nutrients can be provided to them with 10 kg quantity of the same food. So this kind of a change is being done to ensure that we can provide the required food to the animal. Then there are herbicide tolerant plants, or HT variant crops. So in case of HT variant crops, what we see is that they would be able to resist herbicides. What are these herbicides? Herbicides will be used in the farms, will be used in agriculture, so that any unwanted growth, any weed that would be growing in the given crop field, that can be killed. So if you have a herbicide resistant crop, then it means that this crop will be able to resist the herbicides, meaning that they will not be affected by herbicides. All right, they will not be affected by herbicides, and they can grow very well without getting affected. So otherwise there are chances that the herbicide would have affected the crop as well, and it could have reduced the productivity of the crop. Then the use of biofertilizers. So in case of biofertilizers also, for example, let's say, in the biogas plant also, what we discussed is that biogas plant, you are getting all the biogas from the required waste, but the leftover waste after biogas extraction has happened, that leftover waste can also be used as a biofertilizer. So that's why this is also an agricultural application, where we are using the leftover of agriculture as biofertilizers. Then also lastly, improvement in floriculture. That even so, when it comes to agricultural applications, the agricultural applications are not only restricted to growing food crops, adding nutrients or adding productivity and so on, but we see that in the cash crops, in the commercial crops also, they are very very useful. In the commercial domain also, they are very very useful. So one such very important area is floriculture. Because in floriculture, see, when it comes to flower cultivation, when you're, let's say, for your garden, if you're buying flowers, or for your home if you're buying flowers, then there are certain expectations that you also have. You would want that the flower looks beautiful. You would want perhaps that there's a very wonderful smell coming from the flower. And at the same time, you wouldn't want that the flower only lasts for a couple of days. You would want that it should last for, let's say, 10, 15, 20 days if possible. So there are all these expectations that we have from the flower as well. So that's why this floriculture industry also will be using these kind of things. Similarly, we see that recently, last year and last to last year, we see that GM rubber was also in news. GM rubber was in news, and in the case of GM rubber, this is a kind of rubber that we want to grow in the northeastern states, especially in especially in Assam and in Arunachal Pradesh and parts of Meghalaya. So that central part of northeastern India, this is basically where we want to grow GM rubber. So now, for GM rubber to grow in these areas, there are certain qualities that it needs to have, because largely what we see is that rubber is grown in the southern part of the country, where the temperature ranges are moderate. It never goes very high, never it drops very low. So that's why those kind of conditions are good for rubber growth. Whereas in northeast, that's not the case. In northeast, the temperatures can go down uh, uh, to very cold temperatures. So that's why we need to ensure that if we want to grow rubber there, they should be able to survive the cold winters. So that's the modification that has been done, so that they can survive these kind of conditions, which otherwise do not exist in the southern parts of India, where we grow rubber. So that's why we see that in all these areas, we are looking for, even that's why not only food crops but also commercial crops. Look at this question. This question was asked in 2011. Please read the question. I'm launching the poll for you. All right, for this one, I'll not launch the poll. All right, for this one, I'm not launching the poll. I want your answers in the comment. I'll give you the reason why I'm not launching the poll. Your answers, please, in the comment. All right, so many of you are saying C, many of you are saying D. All right, so let me give you the answer. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? First, to enable them to withstand drought. We discussed this yesterday, that we try to uh, make changes in a manner that they can survive in low water conditions as well. So this is anyway something that we do. To increase the nutritive value of produce, we discussed this as well, that we do this. Fourth, also, if you see, to increase their shelf life, we do this. We discussed this yesterday as well, that we can increase the shelf life of the food as well as of the seeds. We can do that. We can do both. Now coming to statement number 3. All right, so 1, 2, and 4 for sure are correct. So A and B can't be answers. All right, A and B cannot be the answers. So 1, 2, and 4 definitely correct. Third statement is to enable them to grow and do photosynthesis in spaceships and space stations. Can this be done? The answer is yes. Today we can. There are certain experiments that have been carried out. For example, one such experiment was carried out in the year 2019 by NASA, and I think in 2022 they have again uh, done that in International Space Station. So that's why now it is possible. Now we know that it is possible. But look at when the question came. Question came in 2011, and again read the question back carefully. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? The question is saying have been. The question is not saying can be. If it, let's say, was a can be question, then in science, can be anything. So then you close your eyes and mark all of the above. Then you don't have to worry. But it says have been created. So have been means that unless it has been done until now, you can't mark your answers, your options correctly. All right. So unless you know that this already has been done, you cannot mark this as correct. So that's why in 2011 we had not done it yet. In 2011, photosynthesis in space was not something that we had done. It was still a part of research. It's still a part of study. So that's why if the question is being answered in 2011, the answer is C. If the if the question is being asked today, the answer is D. That is the reason why I did not launch the poll, because it's difficult to say. I mean, so that's why every one of you who marked C or every one of you who marked D, just add 1 point to your leaderboard score. All right. So this is it. So that's why, I mean, the language many a times matters. Language many a times would matter. And that's why, I mean, many a times these kind of small changes UPSC does, and they become happy about it, that look, we changed the uh, language in a manner that the students will get confused. So they shouldn't do that, but they do this all the time. All right. So that's basically how this question had to be answered. So that's why, always remember, I mean, the language of the question always matters. What they have written, what they have mentioned, always will be very, very crucial for you to mark the things correctly, to answer the things correctly. All right. So that's why the correct answer here could be C. I mean, if you're looking from the official answer perspective, from the perspective that it was asked in 2011, it will be C. Today being answered, the answer will be D. All right. Next, we move on to white biotechnology. Now, white biotechnology basically means that we are using biotechnology in industries. So now in industries also there are multiple applications. For example, let's say we discussed about biofuels. So the same biofuels that we discussed as agricultural application can also be counted as industrial application, because it's an industrial fuel as well. It's a industrial process making this from food crops. So that's why it will also be considered as white biotechnology. Similarly, there are other processes as well. For example, let's say the process of fermentation, which is used in various different ways in the industries, in making the biofuels. There are so many ways in which fermentation as a process would also be used. Then also we see that even to enhance oil recovery, we can make the use of biotechnology, and we discussed anyway about the biofuels. So there are various different processes. So for example, there is something called as biosorption. So biosorption is what, if you break down the word, you can say that in a manner this is biological plus absorption. So that's why we are calling it as biosorption. So now, biological absorption, meaning that you have something which can absorb some material, right? You have, let's say, an organism, a microorganism that can absorb the material. So this is basically what we are talking about, that to bind and concentrate heavy metals from even very dilute aqueous solutions. So in this particular scenario, what we basically see is that, if you see here, the metallic ions, they can be absorbed. They can be absorbed by the microorganisms, or maybe some smaller organisms. They can feed upon these metal ions, and these metal ions then can be removed. So in a manner, to decontaminate in a area, we can use it, or also for something called as biomining, something similar can be used. So biomining, so biomining is also very similar, has also a very similar approach, where we are trying to use the microorganisms in a manner that they would absorb anything that is there in the surrounding. All right. So for example, let's say, to leach out minerals, or let's say even in oils also, we see that something like this can be done. In metal absorption also, something like this can be done. So in all these cases, we see that the microorganisms or any such organism will be used, and they would be the ones to absorb everything, and then from there we will try to extract them. Especially when it comes to very dilute solutions, or let's say dilute environments, that is where the normal industrial method becomes very costly. Normal industrial method is cost effective when, let's say, very small quantity. Then the same industrial method can become very costly, and then there is no use for you to take out those material, because you are basically using, I mean, you are spending more money in getting something that will give you lesser money. So then it does not make sense. So in those kind of environments especially, we see that biomining as a process will be used. Similarly, in case of biomining, a kind of biomining is also called as phytomining. Phyto, just remember that wherever you see this term phyto means that we are talking about plants. Phyto means that we are talking about plants. So that's why when plants can be used for any kind of biomining process, we'll say that it is, that kind of biomining is phytomining. All right. So many a times what happens is that roots of the plants can be used. Roots of the plants can also be used. So let's say you have soil which has some concentration of copper, dilute concentration of copper. So then in that case these copper ions can be extracted, they can be absorbed with the help of roots of a certain tree or plants that are growing in that area, and then from those roots we can actually extract copper. Those roots, many times what we can do is we can burn the roots, and from the ashes we can collect copper. So in this manner we see that even the process of phytomining, which is a kind of biomining, is used. Then after that we have blue biotechnology. Now blue biotechnology is simple. Whenever anything related to water, anything that is related to water will be called as blue biotechnology. Now, in case of blue biotechnology, what we basically see is that first and foremost, a very, very important example here is oil zapper. Example is oil zapper. Now this oil zapper is nothing but a combination of 5 different bacteria that can help against oil spills. All right. So whenever there are oil spills, we see that oil zapper can be used, because oil zapper, a formulation made of 5 different bacteria, this oil zapper will be taken to the site of contamination wherever the oil spill has happened. It will go there, it will start to eat upon all the hydrocarbons and break them down. So what happens is that this hydrocarbon will be broken down into carbon dioxide and moisture, or carbon dioxide and water molecules. So that breakdown basically will ensure that whatever oil spill had happened, that can be slowly eaten away with the help of oil zapper. So that is basically what we see. So oil zapper, that's why we are saying that it converts hydrocarbons into carbon dioxide and water. Then there are certain transgenic aquatic organisms as well, wherever required and whatever be the requirement, accordingly we can create those aquatic animals and plants with modified properties also. Then the next topic is the use of biotechnology in the environment. So in the environment, what we call as gray biotechnology. Now, in case of gray biotechnology, we see that the process that we are using here is called as bioremediation. All right, the process is called as bioremediation. So bio, because living organisms, remediation is basically cleaning up. So when you are trying to clean up the environment with the help of any biological source, whether plants or animals or microorganisms and so on, then in that case we say that it is bioremediation. Now, in case of bioremediation, we see that there are two types of bioremediation. One which is called as in situ bioremediation, and another one which is called as ex situ bioremediation. So you might have heard of these terms: in situ conservation, ex situ conservation. These kind of terms are used in the environment and ecology classes, right? So in situ, the meaning of the term in situ is at the site. All right. In situ means at the site, and ex situ means away from the site. So in situ means at the site, ex situ means away from the site. So in this particular scenario, what we see is that in situ and ex situ bioremediation techniques, these are used. Now, when it comes to these techniques, first one by one we'll try to understand them, and there are various processes that are classified under in situ and ex situ. So first coming to in situ. So in situ, when we are saying at the site, meaning that we are trying to treat the waste at the site of contamination itself. So wherever contamination has happened, we are trying to treat the contamination at that particular site itself, and that is called as in situ bioremediation. Now, in case of in situ bioremediation, there are multiple different types of processes that are used. So, for example, we have biosparging, we have venting, we have bioaugmentation, we have bioculture, etc. So, there are multiple different kinds of methods that we can use. Biosparging and bioventing are the two most common methods that we use here. So, let's try to understand this first. That what do we mean by biosparging and what do we mean by bioventing? Because both the processes are fairly similar, just that there are certain small differences between what is biosparging versus what is bio venting. So you have biosparging, sorry, you have biosparging and you have bio venting. So now what happens here is, just understand this, that let's say we have layers of soil. Suppose that you have layer of soil, and below the layer of soil, you know that ultimately water table would begin, correct? Water table will begin after some depth. So now in this particular scenario, let's say when we are considering this top layer of the soil versus this layer of the soil, this layer of the soil versus this layer of the soil. Now, between the top layer and the bottom layer of the soil will have more moisture, right? It will have more moisture because it is nearer to the water body, it is nearer to the water table. So that's why it will have more of moisture. And when it comes to the top layer will not have that much of moisture because it is also in contact with sunlight, and a lot of evaporation, etc., will also keep on happening. So that's why it will not have as much moisture as the bottom layer would have. Now in these two areas the microorganisms are also present, correct? The microorganisms are also present in these areas, and any kind of contamination that we would have, all those contamination will also will be taken care of, will be decontaminated by the action of all the microorganisms that we have here. But that natural process is very slow. That natural process is slow. The microorganisms will take their own time to eat upon whatever has come their way, to decontaminate the given area. They would do it, ultimately they would do it, but they would take a lot of time. This is where, with the help of techniques like biosparging and bioventing, we try to enhance their activity. We try to enhance their activity. How exactly do we enhance the activity? By supplying them with more nutrients and oxygen. So when they have more nutrients and oxygen, it means that they'll consume more nutrients, they will become energetic, they'll have extra energy, and that extra energy will be used up in decontaminating the given area. So that's why what we do is that the natural activity that anyway would have happened, we are trying to fasten the process. We are trying to fasten the process by adding extra nutrients and adding extra oxygen to the given part of the soil. So this is basically what we do in biosparging and bioventing. The difference that we have, the difference that we have is that in one case we are going here, and in another case we have to inject the oxygen and the nutrients to the bottom layer. So now that's why when you are going to the bottom layer, you basically have to exert more pressure, correct? You have to exert more pressure. And when you are at the top layer, you don't have to exert that pressure, correct? You do not have to exert that much amount of pressure. So carrying out the same process in the upper layer is easier. Carrying out the same process at the lower level is a little more difficult. So the first one that we have, the at the top layer, what we have is bioventing. This is where we have bioventing, and this is where we have biosparging. So that's why when it comes to biosparging, in case of biosparging, we do it in the saturated zone. So saturated zone is this zone. That's why saturated. Why saturated? Because it is near the water body. It is near the water body, it is near the water table, and hence it has more moisture, and thus it is called as saturated. Because why saturated? Because it can't take more moisture. It has enough of moisture already. It can't take more moisture. So you say that it is saturated zone. Whereas unsaturated zone, where there is scope for adding more moisture. So that's why in case of biosparging, we see that we will be using high pressure. Air or oxygen and nutrients are injected at high pressure to increase the biological activity of the indigenous microorganisms to enhance the decomposition activity. So this is what we do in biosparging. So just to repeat again, you have top layer called as unsaturated zone, you have bottom layer called as saturated zone. So this is called as unsaturated zone. This is called as unsaturated zone, and this is called as saturated zone. This is called as saturated zone. All right. So you have unsaturated zone and saturated zone. So in the unsaturated zone, injecting will be easier. So we will be doing that at low pressure. We'll be doing that at low pressure. So here we are doing this in the unsaturated zone. All right. Unsaturated zone. And in this particular scenario, we see that we will be inducing oxygen or the nutrients at low pressure. So that is basically the basic difference between what is biosparging and what is bioventing. So in the decomposition activity, in decontamination, for example, the decontamination of water bodies can also be done with the help of biosparging. The decomposition of saturated soil also can be done by biosparging. The decomposition of unsaturated soil will be done by bioventing. So that's the basic difference between the processes. Then after that we have XC2 bioremediation techniques. Now in case of XC2 bioremediation techniques, just remember that when it comes to these two, uh, this technique, we are now saying that the contamination will be taken away from the site. All right, contamination will be taken away from the site, and this is where the treatment of the contamination will be done. So first such technique, for example, is land farming. So what we do in case of land farming is that we are taking the contaminated soil and it is spread over a prepared bed, and periodically tillage of the soil is done. So tillage of the soil, the tilling of the soil, what we basically see is that tillage will be able to wake up the microorganisms. The microorganisms are sleeping very peacefully in this soil, and you wake them up by the tillage process. You wake them up. So that's why their activities will increase. They will not keep on sleeping all day long, and hence we see that and that's why the decomposition activity can be enhanced, and hence they will be then functional, and they will be able to do the decontamination process faster. So that's why tillage is a very very important process in any kind of agriculture. Then after that, we have biopile. Now, in case of biopile, it is a more specific method where various techniques like land farming and composting techniques will also be used. So now in case of biopile, each and every property of the soil, each and every property of the soil is perfectly maintained. So for example, what is the required moisture level in the soil? What should be the pH level of the soil? Whether it should be acidic, basic, how exactly it should be. So that will be maintained. The heat content of the soil, water content of the soil, we already discussed. The oxygen level of the soil, so the nitrates and the phosphates that are there in the soil. So each and every property of the soil, each and every nutrient required by the soil, everything will be properly maintained, so that it can be used for any required purpose. The soil can be then used for any required purpose. So that's why it takes a lot of time preparing this biopile, and then this biopile would be sent. It could be sold in the market as well. It would be sent everywhere. So this biopile, then what we see is that this biopile can take a lot of time. It can take even 3 to 6 months of time for the preparation of this biopile. But essentially, we will ensure that each and every property, it can be properly maintained. So moisture, heat, nutrients, oxygen, pH, they are all controlled to enhance the biodegradation process. And we see that in this particular scenario, the treatment can typically take up to 3 to 6 months of time. So overall, but we see that biopile will ensure that the quality of the soil is wonderful. Then after that, we have other bioremediation techniques as well. For example, we have phytoremediation. So we discussed that wherever you have the term phyto, it means that we are talking about the plants, correct? We are talking about plants. So in this particular scenario, we are talking about plants and how plants can help in bioremediation techniques. Now, plants naturally, you can say that naturally they are helping us in bioremediation, because we know that plants take in carbon dioxide and they give out oxygen. So in a manner, they help in purification of air. But there are certain plants, there are certain trees that go beyond this. So for example, a neem tree is a perfect example of it, where we know neem for its medicinal properties. We know uh, neem for its decontamination properties, and it basically can also absorb poisonous elements and reduces the growth of harmful microorganisms in the given area. So that's why they also help in phytoremediation. So that's why there is an extra amount of work that these plants and trees do. Similarly, we discussed about that how many a times many metal ions etc can be extracted with the help of roots of the plant. So that kind of a process will be called as phyto extraction. That becomes a sub process of phytoremediation. A kind of phytoremediation is phyto extraction, where you are extracting, right, you are extracting the uh, uh, required contaminants from the soil. And especially that goes well for metal ions, because metal ions that are present, or let's say heavy metal ions that are present in the soil, they are not easy to break down. So that's when the decomposers or the microorganisms, they cannot very easily eat upon them, they cannot feed upon them, they cannot decontaminate anything that is related to a metal ion. So metal ions, they have to be largely decontaminated with the help of phytoremediation, because microorganisms will not be able to break the metal ions down. It will be very difficult for them to break down the metal ions. So that's why phyto extraction techniques become much more important there. Then we have mycoremediation. Now, when we say mycoremediation, the term myco is for fungus, and the same fungus that you in plural call as fungi or fungi or fungi or whatever you want to call it. So that's why a safer term for me is fungus, where I don't have to be bothered about how to pronounce this, right? So fungus. So wherever we make the use of fungus in one way or the other, we'll say that it is mycoremediation. So we know that mycoremediation can be used in enhancing the soil properties. It can be used in adding nitrates to the soil. And we see that there are many microorganisms, like different types of fungi, and there are different types of bacteria that can be used in nitrogen fixation. Correct. So in nitrogen fixation, they all can be used. So we know that there are all these examples. So mycorrhiza, for example, is one of them that can be used. So there are multiple such applications that we have when it comes to bioremediation. Look at this question. This question was asked in your paper. Just try. Is the audio and video clear now? Can you just confirm, please? Because I think audio and video got stuck. All right, wonderful. So I'm just launching the poll. I'm sure that you've already read the question. So I think again there was some issue. So because of the issue, I don't even know what poll was marked. So anyway, I'm discussing the answer with you. So in the context of solving pollution problems, what is or are the advantage or the advantages of bioremediation techniques? All right. So if somebody could just give me what was the percentage of students who marked A, B, C, D, if somebody knows. So first statement here is it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. All right, so C is 50% and D is 33% is what I you have written. A is 8%. Okay. So 50% is correct. I mean, C is the answer, but let me discuss this with you. So first statement says it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. So this is perfectly correct. This is what we are doing. That in case of, let's say, biosparging and bioventing also, we discuss the same thing. That although it would have happened naturally, you had the microorganisms present there, but we are trying to enhance the process. We are trying to quicken the process, and that is basically what the first statement anyway says. So that's correct. Second statement says any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Do you remember that we discussed about uh, the extreme words? Look at how many extreme words you have here. Any, readily, completely. So three extreme words in one sentence. Is it even possible that it's a correct statement? Just because of these extreme words, I will anyway say that it's a wrong statement. But why is it wrong? Any contaminant with heavy metals such as cadmium and lead can be readily and completely be treated by bioremediation using microorganisms. What we discussed is that when you have heavy metals, it's better to use phytoremediation there. Correct? Phytoremediation techniques are actually more helpful because it's not easy for microorganisms to eat upon these heavy metals, correct? So that is basically what is happening in this particular scenario, and hence that's why second statement is not correct. Third, genetic engineering can be used to create microorganisms specifically designed for bioremediation. This is also correct. Oil zapper is a simple example. We discussed that in oil zapper you have 5 different kinds of bacteria which have been taken together. So that's the correct answer will be 1 and 3 only. Another question. It's a very simple question. This time I'm sure that more number of students will mark the correct answer. Launching the poll. Please mark your answers quickly. Hmm. So 68% of you have marked A, 11% B, 18% C, and 4% D. So, oil zapper. In what context did we talk about oil zapper? We talked about oil spills, right? We discussed that whenever oil spills happen, you can use oil zapper. Oil zapper will go there. It will eat upon all the hydrocarbons that you have on the surface of the sea or oceans, and you will be able to decontaminate the area. Look at statement A. It is an eco-friendly technology for the remediation of oily sludge and oil spills. This is correct. So you don't even have to look ahead. So B says latest technology for developing undersea oil exploration. It's not related to exploration. We are trying to treat something, right? We are trying to treat the oil spill. Then genetically engineered high biofuel yielding maize variety. I'm not sure why somebody marked this answer. It is the latest technology to control the accidentally caused flames from oil wells. So we are not talking about any kind of flames. We are simply saying that whenever oil spill happen, wherever the oil spill happens, we'll simply use oil zapper there. All right. So that's why the correct answer here will be A. Now we come to some of the specific techniques. So until now, what we have discussed is the various aspects of how biotechnology can be used in various different sectors, in various different fields. Now we come to certain techniques which are specific techniques, or there are specific concepts which are related to biotechnology. So here we'll discuss about what is cloning, what is stem cell, or what is DNA fingerprinting, the issue that we have with GM mustard. So these kind of things is what we are going to discuss in this part of our chapter. Now, before that, if you have any questions, if you have anything from whatever we have discussed until now. The list of radioactive elements I've already shared with the team. I'm sure that it will be given to you as a part of the ARM or something. Oil zapper is, as I told you, just told you, oil zapper is nothing but when you have a formulation where five different types of bacteria are mixed together, which can actually help against oil spills, which can help in oil spills, that wherever oil spill has happened on the sea or the ocean surfaces, oil zapper can be taken there, and this oil zapper will then start eating upon all the hydrocarbon buildup. Land farming is nothing but simply the tillage of the soil. That when you start tilling the soil periodically, that is called as oil. That is called as land farming. Biosparging and bioventing processes is the same. Yes, process is the same, just that more effort has to be put in biosparging, because you need high pressure, you need injection wells that can go deeper inside, and hence biosparging is little costlier also compared to bioventing as a process. Do we have any bioremediation for plastic waste? Not exactly, but what we have been doing is that we have been creating genetically engineered microorganisms that probably can also feed on plastic. All right. So we have been trying specific methods of biotechnology there to make those microorganisms. Otherwise, naturally, we don't. The difference between biosparging and bioremediation? No, there's no difference. Biosparging is a kind of bioremediation, right? Bioremediation is any way that you clean up the environment, and biosparging is one of those ways in which you clean up the environment. The difference between phytomining and phytoextraction, you can simply say that phytoextraction is a kind of phytomining. Let me just write here. So if you see here, it is written that it is a sub process of phytoremediation in which plants can remove the dangerous elements or compounds from the soil. So that's why when they are extracting from the soil, when plants are extracting from the soil, you are saying that this is phytoextraction. So you can say that it's a sub process of phytoremediation. In situ means that we are present at the site. Yes, it means that, not we present at the site. What it basically means is that wherever the contamination is, you are treating the contamination at that particular point itself. All right, you're treating the contamination then and there itself. So that is why it is called as in situ. In situ and ex situ. In situ and ex situ is simple, that we are talking about how, or rather where the process is carried out. I mean, where exactly are we treating the waste? So that basically becomes uh, so if let's say you are doing it at the site of if you're doing it at the site of the contamination itself, it is called as in situ. Otherwise you are saying that it's ex situ. What is rDNA? rDNA is nothing but the recombinant DNA. When we say recombinant DNA, what we are doing is that we are combining two different types of DNA. So when we do that, we can say that this is an rDNA. So rDNA is nothing different, just that uh, it's a recombinant DNA. If H2S is released as a byproduct into the environment, how does it harm the environment? So there are a lot of things. I mean, for example, it can cause difficulty in breathing. I mean, for starters, it can cause difficulty in breathing, some kind of infection of the nose and throat, etc. It can cause that. And then if the concentration is higher, then in that case it becomes much more difficult, because then it can lead to a lot of issues. It can in larger it can even induce a coma in students, in patients, if the concentration levels are very high. So that is basically what we see when it comes to hydrogen sulfide. So otherwise there are smaller effects that hydrogen sulfide might have in lower concentration. So that's why wherever in the industries hydrogen sulfide gas is a byproduct, in many of the industries, uh, in fertilizer making, for example, in even in agricultural farm we see that, in textile industry we see that. So in all these industries, wherever hydrogen sulfide is a byproduct, there are these issues that can happen. Isn't it ethically wrong? What exactly are you talking about? I'm not sure. Can you tell me the context in which you are saying this? Can biosparging and bioventing be useful in removing heavy metals such as lead and cadmium? Not really. Uh, very in a very limited way. Not exactly. Not completely. Due to high pressure, water evaporates before the boiling point or do not evaporate after boiling point. See, actually both can happen, depends on how the vessel actually has been created. So in nature, if we do that, uh, the other ambient conditions also matter. In the industrial processes, for example, the process like when we use it in a nuclear reactor, there after boiling point also it will remain liquid. But in there are certain cases as well where before boiling point also it can evaporate because of the pressure changes. So it actually depends on the other ambient conditions, and it depends on how exactly are we doing it, or what kind of a system is being used there. Can you give some examples of ex situ experiment? It's not an experiment, it's a process. So please don't call it experiment. And we just discussed, I mean, biopile technique. For example, let's say if you go and buy some soil from somewhere. So that prepared soil that they're giving you, that prepared soil is nothing but biopile that you get. Similarly, the basic farming techniques also, they are using XC2 bioremediation, right? So next we come to our next topic, that is cloning. Now, when it comes to cloning as a process, what we say is that cloning is a process of asexual reproduction in which the offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. Now, understand this, that what exactly do we mean by this process of cloning? So, when it comes to cloning as a process, for the first time, cloning as a process was carried back in 1997. And this example that you see right here, this was the first such example. Dolly the sheep. All right. Dolly the sheep, where the sheep was produced with the help of cloning techniques. Now, what do we mean by cloning? Cloning means to produce an exact replica. All right. When you produce an exact replica of something, you would say that you have cloned it. Correct. You have carried out the process of cloning. You will say that you have carried out such process. All right. So now, in case of cloning, first and foremost, why cloning? And second is how cloning. Correct. So why cloning? The answer is that in many cases we see that there are certain properties in these animals that we want to replicate. We want those properties, so that's why we want to replicate them. We want more of the same variety, we want more of the same quality. So whether it is in food production, whether it is in milk production, whether it is in farming, whether it is about anything in which we see that the animals might become useful for us, and there are certain animals which have shown those properties, who are genetically, let's say, so to say, they are genetically superior. They can they can fight diseases better. They have they give us better milk production in any way. All right. So if we see these things, then in that case we might carry on the process of cloning. So we are saying that it's a process of asexual reproduction. Now, why do we say asexual? Because when it comes to the two types of reproduction, one is sexual and one is asexual. Sexual reproduction is where you have a mother and a father, meaning that between a male and a female cell, the process of fertilization will happen, and fertilization will also mean that genetically these two are mixing, that there are genes from the mother and the father which are mixing, and then there will be a unique offspring that will be produced out of that process of sexual reproduction. So largely what we see is that almost all the higher plants and animals, including the humans, uh, all these basically reproduce via the means of sexual reproduction. Whereas when it comes to asexual reproduction, there the genes are coming only from one parent. All right, genes come from only one parent. So in a manner you can say that you are producing a replica of the parent. And there are, again, there are multiple techniques in nature also that we see. For example, a simple technique of budding, what we see is that a small bud will be formed on the body of the organism, and this bud starts to grow, and after a point when the bud has grown, it will detach from this particular organism. So then in that case, this is an example of asexual reproduction, because the birth of the offspring happened from the same one single parent. So same happens in cloning as well, that in case of cloning also there is one single parent, and from that single parent itself, we see that the process of reproduction is carried on. Now, cloning as a process is a very difficult process. It's a very complex process and a very unstable process. So that's why we have seen that there has been limited success in the beginning, and it's only of late that we have started getting more out of cloning. We have started getting more success in the process of cloning. Initially, it has been very, very difficult. So, for example, the first such technique was Dolly. But maybe before Dolly, it was Shaktiman as well, and there are other English films also where the processes have been much more simpler. That you put one person in one box, and you see that another person comes out of another box, and that person is a clone of the person that you had put in the box. I wish the things were that simple. that you could put one person in a box and another person comes out of another box and becomes the process of cloning. So it's not it's not that easy that the process of cloning will be carried out. So what we see here is that you have to carry it out with the help of genes, and that process initially, for example, has been very very complex, and we did not get a lot of success. So for example, even this, the first successful example, just a trivia, KBC, can you tell me how many attempts it took for this clone to be produced? Okay, I see different numbers: 86, 17, 36, 7, more than 50, 100, 3. So let me give you the exact number. It took a total of 277 attempts. Yes, Amrit, you are correct. 277 attempts. So after 277 attempts, we were successful. How many attempts do you get in this particular examination? So I'm not going there. So it took 277 attempts for us to be successful finally, uh, in case of the first cloned animal. So you can imagine the kind of complexity that we uh, face when it comes to these kind of processes. All right. So that's why the first successful cloned animal was a sheep called Dolly, and this is that picture of that particular sheep. Now, in case of cloning in India, we have seen that there are certain examples in India as well. For example, the first Indian example was Samrupa. This was 2009, and it was world's first cloned buffalo. The calf did not live for very long. Only just after 5 days, it succumbed to lung infection and died. After that, Garima was the second example. The second cloned buffalo lived for like 2 years, but again did not. After that, it died. And then after that, CIRB Gaurav has been the third example, where we see that it has been much more stabilized. There have been recent examples as well. This was an example in 2020, and this is an example this year itself, 2023. So in 2020, we carried down from the same CIRB Gaurav or Hisar Gaurav, cloned buffalo, a recloning of it was done, and 7 cloning of M29 Murrah bulls were done. So this is basically a picture of that. And this on the other side of your screen is the most recent example. Do you know about this? Anyone who knows? This was this year in March 2023. Yes, Swati, you are perfectly correct. So this is basically the name given to this clone was Ganga, and this is a kind of a gir cow. Now, in this particular scenario, why gir cow was chosen? It was mainly because of a natural immunity against diseases that we have seen in gir cow. So that's why we here the basic idea was that instead of uh, relying more on the foreign breeds which are very difficult to survive in India, and we have to take a lot of care for making sure that they survive. Let's do a cloning with indigenous varieties, and because these indigenous varieties, uh, they have a natural resistance towards the tropical diseases, the diseases that grapple India or other parts of similar regions. So that's why the cloning process was carried out with a cow of indigenous variety. So that's why we see that there have been all these examples in India of animal cloning. So to understand what exactly do we do and how exactly the process of cloning is carried on, just try to understand with the help of this particular picture. So first and foremost, what we have to do here is that we have to isolate something called as somatic cells. All right, we have to isolate the somatic cells. Now, these somatic cells, when we are saying that we want to isolate the somatic cells, what we mean by somatic cells is any cell in our body which is non-reproductive in nature. So for example, in humans, there are two cells which can be called as reproductive cells. You have the egg cell in the females and you have the sperm cells in the males. These are called as reproductive cells. Apart from these two cells, every other cell in the body, they are called as somatic cells. So the first process here in cloning would be that we isolate the somatic cell. Once we have isolated the somatic cell, we have to find an egg cell, because ultimately the growth of the embryo happens in the egg cell, correct, with the help of the egg cell. So that's why we need that egg cell. Now look at the next thing that we do, that we take away the nucleus. We take away the nucleus of this egg cell, and we add the somatic cell or the somatic nucleus to the egg cell. So that's why now you have the egg cell with the somatic nucleus. Can you think of the reason why the nucleus? Why are we removing the nucleus from the egg cell and we are adding the nucleus from the somatic cell? Can you think of a reason? Can you tell me what the reason could be? Okay, Tagdis, you are correct. Yes, Deepika, you are correct. Yashpal, correct. Nitesh, correct. Swati, Varun, correct. Shaili, correct. All right. So I see that you are getting the picture. So basically what we are doing is, and what we know about nucleus is that nucleus contains DNA, right? We discussed this, that nucleus contains the DNA, and the DNA that is contained inside the nucleus has all the specific characteristics of this individual. All the specific characteristics of the individual are inside the nucleus. So that's why if we want the characteristics of only this person, then in that case we need to take the nucleus out of this person and ensure that there is only this nucleus. Otherwise what happens in a natural sexual reproduction is that the nuclei from both the sperm cell and the egg cell, they would fertilize together, and there will be a mixing of genes that will happen from both the different nuclei. Whereas in this particular process, we don't want the other nuclei to be functional, to be participant. So that's why we have to take the nucleus from the somatic cell and we remove the nucleus from the egg cell. Now, once we have this egg cell with the somatic nucleus, we see that with the help of shocks or with the help of some chemicals, we produce something called as a blastocyst. This is called as blastocyst. So blastocyst is where you have a bunch of undifferentiated cells, almost let's say 115 number. You have undifferentiated cells, and they basically are the ones that will be used. Now the last process here could either be that if we want birth of a new individual, then in that case it is called as reproductive cloning. If not, if let's say we want to use it for some kind of medical purpose, then it is called as therapeutic cloning. Right? In this particular process, then it is called as therapeutic cloning. So these are the two different ways in which the cloning process would be carried out. So cloning here, that's why it means either it would be reproductive cloning or therapeutic cloning, depending on how exactly are we using this process. So this is what we do in case of cloning. Now, so just to summarize the points, first we take the somatic cells, right? We take the somatic cells and we transfer the nucleus into an enucleated egg cell, meaning that nucleus has been taken away from the cell, and then after that it will be stimulated with the help of electric shock and chemicals to initiate the division. And then we see that we have a single cell that becomes a ball of mass having almost 150 unspecialized cells. This is called as blastocyst. All right, this is called as blastocyst. And then this blastocyst can be inserted inside the uterus of a surrogate mother so that the process of embryonic development can start. Or alternately, if you don't want that, then it will directly be taken to the lab where from you can use this, because this in a manner is where we also can generate stem cells. I will discuss about stem cells a little later. So that is basically the entire process of cloning. So either it could be reproductive cloning, where we are using the embryo for the development into a new individual, and otherwise it can be therapeutic cloning, where we can use it to harvest the stem cells for medical purposes. So how exactly we are doing that, we'll discuss that at a later stage, that what do we mean by stem cells, etc. So look at this. This was asked in your paper. I'm very sure that you can answer this question very quickly. Great. So 89% of you have perfectly given the correct answer. What is the application of somatic cell nuclear transfer technology? So somatic cell nuclear transfer, so this is basically what we are doing here, that we are transferring the nucleus from the somatic cell. This is the process of cloning, so it will be helpful in the reproductive cloning of animals. Now, some of the problems that when it comes to cloning, we have seen that cloning has been tried in animals. We have succeeded in animal cloning. But when it comes to human cloning more or less has been banned all across the world. It has been banned only with very small exception. So because there are a lot of problems that we have with cloning, we know that the things can go very wrong, especially in the society. So for example, let's say the first statement, if you see, that it can undermine society's respect for human life, that we might not have the same respect for a clone. We might think of it more like a process or a scientific experiment or something, instead of looking at it as a life form that also exists among us. So that's why we may have this kind of a reservation. It might also lead to us looking down upon them, and in a manner you can say that they can be treated as secondary or even as slaves. Then after that, it can also affect the social institutions such as marriage and family, because even people who are single, they can think that why do I need to get married. uh, Especially, let's say, if somebody is a fan of themselves, they'll think that no, my child should be exactly like me. So that's why I'll carry on the process of cloning, and I'll make sure that my son or my daughter is exactly like me. So that also can be done. That can be done with the help of cloning. So that's why, and this then breaks down the societies. I mean, it's not uh, a thing to joke about, maybe, that it might lead to a very different kind of a change that may happen in the society, where people do not give respect to the institutions of marriage or starting a social life, starting a family, etc. And that can have a negative impact on the society. It may not look so for that individual, but overall, when we are looking at the society, it may have a negative impact on it. Then after that, it may even make global security concerns. It can create uh, global security concerns, because ultimately what may happen is that whether terrorist organizations or even governments, they might start working on the process of cloning, so that 15-14 years later they have a cloned army, and then they don't have to use their own army. Instead of that, they will be using clones in the wars. So that can be dangerous. Then also, there is a concept called as designer babies. Now the concept of designer babies basically is about the fact that many a times what we may want many a times what we may want is that we want the child to be born with specific characteristics. So if we want that, then in that case we might want those genes that we need in the child. So then we are exactly designing that child, we are exactly designing the child according to our wishes. So that is the concept of designer babies. Again, this can be this can have a very negative impact on the things. And then one more problem, the ethical issue that we have, is mainly the problem related to the work. For example, in case of, let's say, when we talk about therapeutic cloning, then therapeutic cloning means that the embryos that have been produced, these embryos will now be used to harvest stem cells, to generate stem cells from there. So ultimately, or let's say, even if it is used for some kind of experiment, some kind of study, then in those cases as well, you in a manner are killing a life. If let's say out of that cell, uh, if the birth of a new cell is not supposed to happen, if or of a new individual is not supposed to happen, then in a manner you are killing a life. And that's why many cultures also believe that the life does not begin after the child is born, but rather at the conception, at the time of embryonic development, the new life has started to begin. So that's why from that perspective it is an ethical issue. And also commercialization of embryos can happen. Commercialization of embryos can also happen because of this. So there are all these issues as well. So I'm just writing it here: commercialization of embryos. All right, commercialization of embryos is also possible. Now, one more thing that we should understand is the difference between cloning and surrogacy. Because whatever process we have just defined, it is nothing as compared to surrogacy. I mean, nothing comparable to surrogacy. Because in case of surrogacy, just understand that the process of surrogacy is still close to sexual reproduction. Because still the fertilization will happen between a male and a female cell, right? And then the embryonic development will happen, and thus the process is different. So that's why first in the process we know that in cloning a somatic cell is being taken, whereas when it comes to surrogacy or in vitro fertilization, we see that the process will be similar to the normal process of birth. Then the uniqueness of progeny. In case of surrogacy, because there is a mixing of DNA happening, that's why the progeny or the offspring will be unique. Whereas in case of cloning, we know that it's an exact replica, right? It's an exact replica of the single parent from where the child has been taken. Then in case of lifespan, there is a big challenge that in case of surrogacy, any child born out of surrogacy would lead a normal healthy life. There will be no issues as such. But when it comes to cloning, we already have seen the kind of instability in the process of cloning, that the cloned animals, they have not lived for very long. They have to be kept in very uh, specific conditions. They can get infected with diseases very easily. So there are multiple such problems that are there with animal cloning. So that's why the lifespan of a lot of animals, the cloned animals, they have not been that high. And then lastly, the ethical issues. The ethical issues with regards to in vitro fertilization, uh, beside beyond a certain some legal barriers, we see that there is no other issue. Whereas in case of cloning, we understand that there are so many concerns, and that's why we see that more or less it is not allowed, and that's why there is so much of restriction that has been put on cloning in humans. Now, one more concept before we take a break. This is about the stem cells. Now, first and foremost, when it comes to the concept of stem cells, understand what stem cells are. So, stem cells, you can say that these are unspecialized cells. These are unspecialized cells from where all the cells in our body will be formed. So, before a cell, because what we know is that let's say when you talk about our body, then in our body there are multiple different types of cells, that we have red blood cells, we have white blood cells, we have nerve cells, we have skin cells, and so on, correct? So there are so many different types of cells that we have in our body. Now all these cells have specific functions, correct? They all have specific functions and they carry one function or the other. But before they become specialized, before they start to perform that particular function for our body, they were unspecialized at some time and they developed into this specialized cell. So that unspecialized form of a cell where every cell is born from, that is called as the stem cell. So for example, let's say if you think of an embryo, so that the embryo just after fertilization does not have all the body parts that we do, correct? But gradually over the next 9 months, we know that the fetus will keep on developing and each and every body organ will start to develop from there. Similarly, in case of a cell also, first there will be these unspecialized cells called as somatic cells, and these somatic cells ultimately will be specializing into various different types of cells. So that's why whether it's a nerve cell or a muscle cell or a skin cell or whatever kind of cell it is, all these cells will be ultimately formed from stem cells. So that's why these stem cells, they have an ability to uh, develop into anything. They can develop into anything, they can develop into any kind of cell. All right. So that's why stem cells become so important. That's why we are discussing about stem cells so much, and there is so much of research that is going on in the process, in the concept of stem cells, because we are trying to understand how many ways can we use stem cells. We are trying to understand there are so many ways where stem cells can become useful. So that's why stem cells, they become the raw material for all the other cells. All right. They become raw materials for all the other cells, and all the cells which have their specialized functions, they would have developed from these stem cells. So overall, this is basically what we see in case of stem cells. Now overall, they have the ability to develop into different specialized body cells. So that's why we know that the stem cells, they can become anything. They have the quality that they can become all these different types of cells. Now when it comes to stem cells, right, when it comes to stem cells, understand two ways of stem cells. That first and foremost, what are these properties? Why exactly are we so excited about stem cells, and why are there so many researches that are going on with regards to stem cells? First is because of their ability of self renewal. All right, their ability of self renewal, that they can renew themselves, meaning that before they start to specialize, before the stem cells they start to specialize, there would be multiple cycles of cell division. All right, there will be multiple cycles of cell division, and these multiple cycles of cell division will ensure that there are multiple copies of stem cells that are available. And then after they have completed their all these cycles and there are multiple cells, stem cells available, this is where they start to specialize. So that's why they go through numerous cycles of cell division while maintaining the undifferentiated state. And this is where we see that if we have to use stem cells, it also means that for us also there will be multiple copies of these stem cells which will be available. Then their ability to specialize, that we know that they have the capacity to differentiate into specialized cell types. So all these specialized cell types they can basically develop into. So that's why we are talking about their ability to specialize. So in all these cases, we know that because of these two specific properties, we see that stem cells become very, very useful for us. So now, based on this, there are various different types of stem cells. So what are these types? Just try to understand this. Very simple. There are three types of stem cells. We have totipotent, pluripotent, and multipotent cells. Right? Totipotent, pluripotent, and multipotent cells. So totipotent cells, basically you can say in a manner, these are cells which can specialize into any kind of cell. All right, they can specialize into any kind of cell. So that's why we are saying that they can specialize into all cell types in the body. They can be specialized into all cell types in the body. And what we see is that the embryonic cells, we discussed that in therapeutic cloning, that from the embryos we can extract the stem cells. So the embryonic cells within the first two cycles of cell division, the embryonic cells within the first two cycles of cell division, they are totipotent. So in case of totipotent cells, we see that totipotent meaning, as I said, that it is derived from the word total. So it means that it can develop into any kind of cell. Right? It can develop into any cell. It can develop into any cell. So largely these are embryonic stem cells. These are also called as embryonic stem cells, because these cells only can be derived from the embryos. So that's why these are also called as embryonic stem cells. Similarly, we have pluripotent cells. Now, in case of pluripotent cells, even these are embryonic in nature. Even these are embryonic in nature, but the difference being that these are seen after the first two cycles of cell division. So they can specialize into, let me write like this, almost any cell. They can specialize into almost any cell. So not all the cells, but almost every cell. So that is basically, they are called as pluripotent cells. Now after this we have multipotent cells. Now multipotent cells have a limited ability to specialize. They cannot really just specialize into anything. So here we'll say that they have limited ability to specialize. Limited ability to specialize. Right? Limited ability to specialize. They basically have two origins. There are two ways in which two areas from where they can be extracted. They either can be adult stem cells. So now they are not being derived from the embryos anymore. All right, they are not being derived from the embryos. So either adult stem cells, or we have something called as perinatal stem cells. Perinatal stem cells. So either adult stem cells or perinatal stem cells. Now adult stem cells does not mean that it is found in the adults. Adult means after birth. Here adult means after birth, because after birth the child would have developed all the body organs. So all the different types of body organs are now in their place. So now in this particular scenario, what we see is that the stem cells also start becoming specific. That for example, the stem cells that are found near the heart, they wouldn't simply grow into a brain cell. All right, they wouldn't grow into a brain cell. Similarly, the ones which are found near the brain, they would not grow into a heart cell. So in this manner we see that there are all these things that we see, and hence they basically have a limited, so their ability to specialize is more limited than the pluripotent cells. So this is what we see when it comes to the different types of these stem cells, right? So lastly, when it comes to the use of stem cells, we see that there are various different ways in which stem cells can be used. For example, something that is called as regenerative medicine. So in case of regenerative medicine, it is where we can use stem cells for any injured part of the body as well, where let's say some kind of, or not only injury, but let's say even because of certain diseases, let's say certain tissues have been damaged, certain cells have been damaged, certain organs have been damaged. So then in these cases stem cells can be used, because we know that the stem cells can grow into these cells, correct? So let's say if due to heart attack sometimes, let's say the muscles near the heart get damaged, so that is where stem cells can be used, so that we can regenerate these muscles at the site of the problem. So that's why it is called as regenerative medicine, or in a manner it is close to what is also called as stem cell therapy. Then it can also be used in drug testing. So in case of drug testing, mainly basically what we can do is that we can recreate a body organ, we can recreate a tissue, and we can test the drugs on it. So before we move on to, let's say, animal subjects or human subjects, first a test on the stem cells can be done. The tissues that have been grown out of the stem cells, there we can first do the test. If that test is successful, then in that case we can move to the animal or the human subjects. So that's why in case of drug testing also, this is basically something that can be done. Then for disease modeling, so we basically, what we can do is that we can create the models of genetic diseases, especially where we can see how exactly the process that is developing from the stem cells, that how from the stem cells the growth is happening. So while the growth of a cell happens from the stem cell, is there any step in the entire process where we see that all of a sudden some kind of disease starts to develop? So that development of the disease can also be found, that can also be understood with the help of stem cells, when we try to analyze the growth of the stem cell into a specialized cell. Then we have stem cell therapy, that in various different types of diseases we can replace the cells with stem cells, we can replace the disease cells with stem cells, and then stem cells perhaps will grow into a normal functioning cell. So it is something that we have tried and we have also succeeded in certain cases. So stem cell therapy, that's why it has become very, very crucial in some of the areas where we did not have a normal medical procedure, or let's say we did not have a medicine or a cure that could save us from those diseases. Now we see that for those kind of diseases we have started using stem cell therapy. Then for basic research also it can be used, that we can uh, find out various ways in which the how the cells function, how they grow, how they die, and that can also give us better insight of the various mechanisms of the diseases as well. Then there is something called as tissue engineering. So in case of tissue engineering, what we know is that it is used to develop the tissues and organs, especially for transplants. And wherever there are any damages because of disease or even due to injury, in all these cases tissue engineering becomes very, very useful. And then again, against neurodegenerative diseases. So neurodegenerative diseases are the ones which happen because of some problem in the neurons, some problem in the, let's say, the cells related to our nervous system. So in all these cases also stem cells can be used so that we can replace those neurons which are causing that neurodegenerative disease with a stem cell, and then the stem cell can be used to solve that particular problem. So that's why there are multiple different uses that we see for stem cells, and there are various ways in which an active research and development has been happening in the field of stem cells. So overall this is basically what we see in terms of stem cells. Now, coming to some of the issues that are related to stem cells. First, the ethical issue, especially when it comes to the use of embryonic stem cells. We discussed about this, that it can create issues, it can create legal issues, ethical issues, commercialization of embryos, etc. is possible. So we have to understand all those issues with embryonic stem cells. The problem with adult stem cells is that they have a limited ability to specialize. So you cannot really use adult stem cells as much as we could have used embryonic stem cells. So we have found a way to solve this problem as well, and this is called as induced pluripotent, called as induced pluripotent cells. So induced pluripotent stem cells is where you are converting converting an adult stem cell into embryonic, adult stem cell into embryonic. So that's why these are called as induced pluripotent, because you are inducing the properties of being in pluripotent in an adult stem cell. So this in a manner solves a problem for us. Look at this question, and after that let's take a break. Read this question. Let me launch the poll for you. Please mark your answers. Hmm. So 1% A, 68% B, 17% C, and 15% D. With reference to stem cells frequently in the news, which of the following statements is or are correct? First statement says stem cells can be derived from mammals only. Only, extreme word, should be wrong. Stem cells, just remember that not only from mammals, but rather from, you can say, all the uh, higher plants and animals. All higher plants and animals. So not only animals, plants and animals. So for example, the tips of the plant, wherever the branches are growing from, the leaves are growing from, the flowers are growing from, the tips, the meristems, the what we call as meristems. So they also basically have stem cells. All right. So that's why both plants and animals. So this is wrong. Stem cells can be used for screening new drugs. We discussed this, that when it comes to uh, a new drug development or a vaccine development, there also stem cells can be used. We grow the tissue or the organ out of stem cell and we test the drugs on that. So that's a screening of drugs. Yes, stem cell can be used for medical therapies. That is why stem cell therapy. So obviously 3 is also correct. So the correct answer will be 2 and 3 only. Right. So this is it. So before moving on to the next question, let's take next topic. Let's take a break. It's 7:53. Let's be back by 8:05. Welcome back, everyone. First, if you have some questions, let me look at them and then we shall proceed. Please explain what is stem cell. So, we discussed the whole thing, but anyway, stem cell, just remember that this is nothing but the raw material. This, you can say that these are the raw material for any kind of cell in the body. This is where any cell that we have in the body, it develops from an unspecialized cell first, and that unspecialized nature of a cell is called as the stem cell. Are these cloned animals sterile or can they reproduce naturally? We often have found that the reproductive nature of the clones are not as good as the normal individuals, the normal animals. We have at least found them to be subfertile, if not completely infertile. Tell us the method of cloning with figures. We just discussed this. Cloning, basically, what we discussed is that you first take, let me go back there. Yeah. So we first would take the somatic cell, and from the somatic cell, the nucleus is taken away. This nucleus will be inserted inside an egg cell where the nucleus has been removed. And then after that, the activation of the cell will be done with the help of electric shock and chemicals. Then you have the blastocyst where you have a bunch of undifferentiated cells. And from there on, it could either be reproductive cloning or therapeutic cloning depending on our requirement. Which type of stem cell is used in the organ on a chip technology? Can we really make human on a chip using totipotent stem cells? Uh, Very unlikely, but yes, we can. I mean, theoretically, yes, we can. And in these kind of cells, will often, I mean, in case of non-human cells, we often see that embryonic cells are also used. I'm not getting the ethical issues with regards last point. Please explain it. Cloning and IVF. So what basically I was saying is that in case of in vitro fertilization, the ethical concerns are not raised as much. There are certain legal aspects of it, yes, and those legal concerns, if they have been taken care of, then there are no ethical concerns as such with surrogacy. But when it comes to cloning, cloning is where we actually are also raising a lot of ethical concerns, that from first and foremost from the societal point of view, and also from the perspective of the fact that embryos are being used for many kind of research, a lot of studies, etc., are being conducted on the stem cells. So that's why these ethical concerns are being raised. How to revise actively and not just passive reading? See, to revise actively, there are a couple of things that you should do. That first, for example, let's say answer writing is a big way in which an active reading habit can be developed. Active revision habit can be developed. At the same time, you can also take, let's say, a blank piece of paper, try to write on your own instead of just trying to just read everything that you have in front of you. So instead of that, just take a piece of paper from your memory, from whatever you remember, try to just write the basic points out there and try to see whether you remember everything or not, and if there are things that are missing from what you have mentioned there. So that will give you an idea about, even in the future, what you are going to miss. Because in general, what we have seen is that if you miss something once, chances are that this is the same thing that you're going to miss again. So that's why these are some of the things that you can do: practicing along with revising, along with writing on your own, writing answers related to those topics. Those are the things that you can actually do. What is this process of multiplication of stem cells called? Is it the same as polymerase chain reaction? Not really, not really. It is simply a process of cell division, the cell division process that normally happens. So it's cell division and nothing else. So cell division will anyway keep on happening in these kind of cells while they are in the undifferentiated form. So polymerase chain reaction is completely different from here. Please explain about cloning in animals and how it became 277. So cloning in animals, just discussed now, that this is where you take the somatic cell and from there on, you are using it for the development of a new individual, and it will be just about cell division that will happen from there on. 277, we were saying that there were 277 attempts that the scientists took before they were successful finally. Can stem cells be used in treating cerebral palsy? Yes, actually, they have been trying. We have seen that with the help of stem cells, there have been a lot of researches where we have tried that experimentally, not with a lot of success, but yes, we have tried. Once explain totipotent, pluripotent, and multipotent. So totipotent, where all the different types of cells can be made from this one cell itself. So then it is totipotent. Pluripotent, where you have small restriction that apart from the reproductive cells, all the other kind of cells can be produced. That is pluripotent. Totipotent, even the reproductive cells can also be produced. And in case of multipotent, we see that there is a restricted way in which the specialization of the stem cells can happen. In simple ways, can it be said that bioremediation, we just activate the microorganisms by some method to enhance them to perform their function effectively? Yes, but not in all the cases. For example, you also use plants, right? In many cases, you also use plants. So there it's not only microorganisms, right? It can be plants as well, or it can be other organisms, it can be even animals. Kindly suggest any book for the subject. For me, it becomes easy to revise. See, book wise, only the NCRTs, only the NCRTs that we discussed. This is not a topic to be read from books. Otherwise, see, many of these pieces of information that I've taken from books which are very highly technical in nature. For example, a lot of things that we have taken in this session, they have been taken from some from class 12th NCRTs, some from biotechnology engineering courses books have been taken in this particular lecture. So you can't really go and read the entire thing. It's impossible for you to read the entire thing. So that's why I would never suggest somebody to go through all these things. Instead, that's why I'm saying that rely on notes and basic reading rather than going into the details. Otherwise, there is no end to these kind of topics. You'll keep reading and you'll never be able to finish the syllabus. From stem cells, sex cells are also formed. The reproductive cells can actually be formed. I mean, when it comes to the totipotent cells can lead to the formation of reproductive cells as well. Blastocyst is nothing but the undifferentiated cell where you have a bunch of cells that have been produced. All right. So what we see is that after the development of embryo starts, we see that first there will be a bunch of cells that will be produced, and that bunch of cells is called as blastocyst. Can we use these totipotent cells to create new organ in the adults? Yes, we can. Which class NCRT is enough? As I told you, class 6 to 8th NCRTs, they are the most important ones. And from class 9th and 10th, only certain specific topics are to be done. Some tricks to be focused. See, depends on your reason to be not focused. So there are multiple reasons why people are not focused these days. One of the most important reasons has been the mobile phone. So if mobile phone is the reason, then do away with your mobile phones. Please do that. It's very, very important. And at least when you are studying, even if let's say it's not possible for you and you're so much addicted to your mobile phones that you can't live without your mobile phone, at least try half an hour. That for half an hour, the mobile phone is kept somewhere where you're not bothered about the mobile phone. You keep it on silent mode or switch off the internet, keep it on airplane mode if possible. Buy a basic phone if possible. One of the things that many do, I mean, even me, I've done something like this, that I've also bought a very small, a 900 rupee basic phone which also has my SIM card, where it does not have internet, it does not have anything else. I only can pick up the phones and send SMSs on it and do nothing else on it. So that also helps me, because otherwise I know that there is so much distraction, everybody contacting you all day long, there are so many WhatsApp messages and emails and all these things happening all day long. So all these things can also be a problem for a lot of us. So that is one. Second, for a lot of students and aspirants especially, it could be a lack of motivation, that a burnout also happens, that you, for let's say the first 3 or 4 months, you pump in a lot of effort, and all of a sudden you see that effort dying. So if that's the case, then there is something else that you'll have to do, that you'll have to start taking breaks, you'll have to start mixing up the subjects, you'll have to start reading things here and there, you'll have to start writing, you'll have to start practicing, discuss with other aspirants, friends, family. So anything that can help you in going into a direction where you are not so monotonous that you feel unmotivated, or let's say you feel disheartened, that is it even possible to clear this examination, and so on. So that's why, depends on the reason why a person is not able to focus. Based on what the reason is, try to first find that reason, that why exactly do you feel that you are not focused, and first finding, after finding that reason, try to think of possible solutions. So it's like to each his own, that for everyone the method could be very different, that for me the method could be very different, for you the method could be very different, that something that helps me to come back and start focusing could not be something that works for you. So you know your methods better than what I would know about you. So just try to find, just sit with yourself for 10 minutes and think what exactly is going wrong and why are you not able to focus, and then come back. It will really help you. Your YouTube lectures also did a great help for me. Okay, the Byju's government exam prep, but even that does not have all the things that are that's required. All right. So I understand that to summarize the chapters, that's a good way to go about it. I mean, the lectures that I have on the Byju's government exam prep channel, uh, but still, I would still say that those are not meant for these kind of areas. I would still prefer that you go and read the class 6 to 8 NCRTs. Yes, for non-science, I understand that it's easy to summarize the things when you look at that particular lecture set that I have there on that channel. But still, I would say that go one step ahead and also read things. Right. So let's move forward. The next topic for us is DNA fingerprint. Yeah. So the next topic for us is DNA fingerprinting. Now, in case of DNA fingerprinting, what we basically see is that this is something where we are trying to identify the complete or partial set of genetic information of a particular individual. Now, overall, when it comes to DNA fingerprinting, first and foremost, why are we using this term fingerprinting? Right. So fingerprinting as a term is being used because it's similar to what happens with a fingerprint. Why is fingerprint important? All right. uh, what is special about fingerprint? What is special about fingerprint? Can you tell me first? Yes, unique to every individual. That's correct. Right. So it's something that is unique to everyone. So my fingerprint does not match with yours. Yours will not match with anyone else. Even if these are identical twins, even then it does not match. All right. So that's why fingerprint, because it's unique. Same goes for DNA. Because let's say when you talk about DNA of any higher animal, including the humans, who are born out of sexual reproduction. So we know that in that case, the DNA, entire set of DNA will be unique. And even in this case also, even if these are identical twins of the same gender, even then there will be small differences in their DNA makeup. So that's why the concept itself has been named as DNA fingerprinting, because this is where uh, we see that everyone's DNA also is very unique to that particular person. So now, when it comes to identification of anything or anything to do with DNA, we see that it is called as DNA fingerprinting, and it has a lot of different applications. For example, it could be about identification of criminals. Now, for example, let's say there is a DNA sample that was collected from the site of the crime, and then there are a list of bunch of suspects that we have. So then the DNA sample that we have found at the site of the crime, that can be used to cross reference with everybody else who is a suspect. So for example, if you look at this, the picture that you have right here, you have the crime, you have the crime scene sample, and you have, let's say, list of three suspects. Can you tell me where, with which subject is it matching? With which subject does the DNA pattern match? Yes, suspect number two, correct. So as you can see that all the three patterns, this one, and this one, all the three match. Because in suspect one, this one does not match, this one does not match. In suspect three, this does not match, this does not match. So that's why only suspect two is where you can see that all the entire set of DNA is matching. So that's why at least you know that suspect number two was present at the site of the crime. You still can't say that uh, that suspect was the criminal, but at least you can zero it down. So that's why we see that in identification of criminals, DNA evidences are used. Then even in paternity claims, or even claim over a dead body after any unfortunate accident where, let's say, the body is not recognizable, we see that in those cases also DNA samples can be used to identify who exactly that person was. And same goes for paternity tests as well, that there also DNA samples can be used to establish the relation between people, the relation between, let's say, a father and the child. So in these kind of cases also, a DNA fingerprint can be used, because there will be certain samples which are genetic in nature, which would have been transferred from the parent to the child, and that's why paternity tests can be useful. Then, to make effective drugs, if you remember, we discussed something called as pharmacogenomics, right? We discussed pharmacogenomics. So this pharmacogenomics is where we are making effective drugs, we are trying to make effective drugs by combining everything related to the physical and the chemical properties of the medicines related to the genetic makeup of an individual, that how exactly genetic makeup of an individual can affect the effectiveness of the drug or efficacy of the drug. So then in that case we can design the drug in a manner that, related to a certain kind of genes, it will be more effective, it will be effective better and faster. So then in that case it is called as pharmacogenomics. Then treatment of genetic vulnerabilities. There also we see that DNA fingerprint can be very, very useful, because with the help of DNA fingerprinting, we can find out the defects in the gene. We can find out what needs to be treated, and then it can be treated accordingly. We can find out the ways in which it can be treated. Then in this case of wildlife management, so in wildlife management, how exactly? In wildlife management, the thing would be about understanding what is causing problems to a certain animal species. That, let's say, we see that there is a critically endangered species, we need to find, and in the DNA itself we will be able to find what are the environmental factors or what are the anthropogenic factors which are leading to the decline in population of this particular animal. So that also can be done with the help of DNA fingerprinting. Not only that, we also collect the sample DNA, especially of the endangered species, especially of the critically endangered species, so that for the future at least we have the reference if at all they are uh, uh, on the verge of extinction, or let's say they are critically endangered. So we see that in all these cases DNA fingerprinting can become very, very useful. But then there are problems associated with it. So for example, something that we discussed in case of cloning as well, the concept of designer babies. Now in case of DNA fingerprinting also allows us to know about genes, allows us to know about the different types of DNA, right? And we know that there are certain genes which are related to this kind of ability or that kind of ability and so on. Now what can happen is, let's say that I want my child to be a great scientist. So if I want my child to be a great scientist, I'll have to find the DNA of a great scientist. Let's say I somehow stumble upon the genes of Albert Einstein. And if I have the genes of Albert Einstein, I would want to use those genes so that I can insert those genes uh, and do the genetic modification while the embryonic development is happening, so that my child, when the child is born, the child has the properties of Albert Einstein. So then I'll be secured in my old age. I'll know that I'm secured in my old age because I've given birth to an Albert Einstein. So it's not a joke anymore. It's something that is actually possible in today's world. There is something which is called as germline editing. Germline editing. Now, in case of germline editing, this is where we are doing genetic modification in the reproductive cells. All right. In the reproductive cells, when we do these kind of changes, these changes become permanent, and these changes will then go on from one generation to another. In case of somatic cells, when we are doing any changes, they remain individual. All right. It will only be there in the next generation, and it's not that it will keep on carrying from generation to generation. But if you do a genetic modification in the reproductive cells, then in that case it is called as germline editing, and this germline editing then can go on from one generation to another. So that's why we see that in case of germline editing, this can be very, very dangerous. So the changes basically in germline editing, the changes in the genes are heritable, meaning that they will be passed on from one generation to another. So then in that case it can become very, very dangerous at times. Also it is unethical also to say so, because then see, there are two ways in which a germline editing can be used. One, if you are trying to treat a disease, that let's say you are trying to treat a genetic disease, then it's great that you have taken care of a genetic disease that could have been transferred from one generation to another. That's great. But on the other hand, you might want certain properties in the child. So then in that case it is wrong, because in a manner it is also said that you are taking a decision for the child without asking a child. That for example, you want your child to be a mathematical genius. What if the child wanted to be a great artist? You did not take that consent. You just decided for that child that this is what I want the child to become. So that's why these kind of cases, these are also unethical, because you do not have the consent and you are just deciding without asking that individual. Similarly, genetic privacy of individuals will also be violated. That we might know about the genes that are present in each and every individual, everyone around us, and then that information can be misused, right? That information can be misused, and we might see that we might look upon certain people just because they have certain genes that for us are so-called inferior genes, that we might say that these are inferior genes, and that's why I don't like that person. So that will become a new way of racism, that will become a new wave of racism. So that also is not good. Then genetic information can also be misused for commercial purposes. That's also possible, that once, let's say, the DNA information about an individual is out, uh, everybody knows about it, then in that case, I mean, especially let's say if the pharmaceutical companies know about it, they can start advertising accordingly. They can tell you that, look, you have these genes mean that 20 years from now you would have cancer. So if you don't want to have cancer in the future, you take these medicines today. So these kind of things can happen, and these are things that very often happen with, let's say, the pharmaceuticals, or in general any kind of advertising. That in so many advertisements, you will see that you would be said that this product has this, because of this will happen, and this is a product that will make you fairer. So it always will basically look down upon your vulnerabilities, and then after that they will try to sell you something on the basis of science. And especially we see that in science things are misused like this, because we don't know science very well. So that's why anything that can be given as a new concept or a new advancement, you probably will buy into it. So for example, uh, a very simple example that I give always in the classroom is charcoal. That today if you see, your face wash has charcoal, your shampoo has charcoal, your food has charcoal, your cosmetics have charcoal, everything has charcoal. Why charcoal, nobody knows, but in some way or the other everybody wants that you should have charcoal, because the toothpaste also has charcoal. So everything has charcoal. Why charcoal, nobody knows, but charcoal. So should we start eating charcoal from tomorrow? Is it so good for us? It's not. And it does not, I mean, and it not, it really makes a lot of difference as well. Just that they will when the advertisements are made, they'll be made in a manner that it will make you feel good about that product even if you don't know about it. And that is what happens with science all the time, that a lot of things that will be explained to you in these advertisements, they are basically, let's say charcoal, let's say, has a positive effect. What will be told to you is that it has so many of these positive effects. So that's the way you sell science when you don't know science. So that is what happens. So, and many a times these are completely new concepts for you. So whatever is explained to you, you will simply accept it. For example, let's say this class, whatever I am explaining, you are accepting it. So you don't know whether I'm explaining it correctly or not, just that I'm explaining it and you are getting it. No, but whatever I'm explaining is not wrong. All right. So this is how we basically know that for commercial purposes these things can be used. So then also creating weapons of mass destruction is also possible. That with the genetic information, what can happen is, let's say for example, against India, some country wants to create a weapon of mass destruction. What do they need? First and foremost, we know that when it comes to this Indian subcontinent, all right, this Indian subcontinent, we know that there is a set of shared genes that has been mixed for thousands of years. For example, on the southern side, the peninsular side, we have the seas that isolates us from the world. Similarly, you have Himalayas on one side, you have the Karakorams on the other side, you have the Thar Desert. So all these areas, the forests, the dense forests of the northeast. So all these are the things that, in a manner, you can say that they isolate the subcontinent. So yes, there are certain genes which are almost, you can say, that would be found in a lot of people who are there in this subcontinent, which perhaps could be different from people in China or people in Europe, that there will be certain genes. Now, if somebody wants to create a weapon of mass destruction against India, they first have to find that gene. They'll try to find that gene first and attack that gene. And then in that case, they can create a weapon of mass destruction against Indians. So that's why we know that all these things are actually possible. And that's why there are so many concerns that we also have with DNA fingerprinting. And that's why we need to understand all these aspects also. Then gene therapy. We discussed about gene therapy. We discussed that uh, depending on what is causing the problem, whether it is an additional gene that is causing the problem, whether it is uh, an absence of a certain gene that is causing the problem, or maybe certain modification in the gene. So depending on that, we can carry out various things. For example, replacing a gene, inactivating or knocking out a gene, or maybe introducing a new gene. So these are three basic processes that can help in carrying out the gene therapy, as we had discussed yesterday as well. But with gene therapy also, we have seen that it hasn't been 100% successful. There are certain issues, there are multiple issues that we have come across. Three of the basic problems that we have seen with gene therapy. One, the short lived nature of gene therapy. Now, many a times what happens is that gene therapy does not work permanently. It works for some time, and then we see that it stops working and it starts to wear off the effects. What basically happens is that if you look at how our body functions and how the cells function, in our body, you might know that the cells always keep on growing, developing, and dying. That cells will be developing, cells will grow into a completely functional cell, and then they will die and they will be replaced with a new cell. And this is something that happens very, very quickly in the body. That, for example, by the time I complete one sentence, almost 10 lakh cells in the body would have been replaced. So, almost 10 lakh cells would have died and replaced with a new cell. So, now what happens is that in these kind of issues, in these kind of aspects, let's say even if a gene therapy has been introduced, what may happen is that the new effects may not uh, go on for very long, and we might see that the body might go on to just functioning as it used to before the gene therapy. So that's why what happens is that the new genes that have been introduced, the body forgets about it. The body might forget about it, and that leads to it being a short-lived nature. And that's why people, many a times, people who have gone through gene therapy, they might have to go through gene therapy multiple number of times from time to time, so that all these issues can be taken care of. Then after this, immune response itself, many a times what happens is that the immune system itself can decrease the effectiveness of gene therapy, because for the immune system also, what we see is that immune system is very simple, that anything that is foreign is an enemy of the immune system. So it does not recognize whether it's a good gene or a bad gene. It just sees that it's foreigner. So if it's a foreigner, destroy it. So many a times what may happen is that the gene that is being introduced itself might get destroyed by the action of the immune system. So the immune system also may not let gene therapy work properly. That also becomes one of the challenges. And then sometimes what we see is that some of these disorders, these are multi-gene disorders. All right, these are multi-gene disorders. For example, heart diseases, high blood pressure, Alzheimer's, arthritis, diabetes. These are diseases which have multiple genes being affected. Means that you have to carry on multiple gene therapies for the gene therapy to work, and that adds to the complexity of uh, or originally a very complex procedure. So that's why we see that this becomes very, very difficult to carry on, and the effectiveness of such a gene therapy where multiple genes have to be replaced might not be as effective as we want it to be. So there are all these problems that we have right now. So that's why we see that only in certain cases gene therapy is being used. It's not that everywhere we are using it. Only in cases where nothing else would work, where there you don't have any medicine, you don't have any cure, any normal medical therapy or a vaccine, etc. In these kind of cases we might want to use gene therapy. And right now also gene therapy is very costly. Both gene therapy and stem cell therapy, they are very costly. So that's why it is not really in the pockets of everyone, not possible for everyone to even think of a gene therapy or a stem cell therapy. Recently, what we have also seen is that the health ministry also has been collaborating with AIMS to ensure that to think of the ways in which they can bring down the cost of gene therapy and other such costly therapies that exist currently. So we see that already the government also has been working on it, but we have to see in the future how exactly things go. Then there is this concept of three parents baby, where there is an extra donor who is required for fertilization. Now understand what exactly is this process and when do we have to use such a process. Now what we see is that there could be scenarios where we see that the mother's egg cell has unhealthy mitochondria. All right, it has unhealthy mitochondria. Now in case of unhealthy mitochondria, what happens is, always remember this, that after fertilization, it's the mother's mitochondria that gets transferred to the child. Father's mitochondria remain with the father. All right, they are not transferred to the child, but mother's mitochondria get transferred to the child. So if mother's mitochondria are diseased in any way, then in that case, those diseased mitochondria will also be transferred to the child. To prevent this, we have the concept of three parents baby, where there will be one donor and the donor would give the mitochondria. The donor would donate the mitochondria, and so that any mitochondrial defect, any mitochondrial disease can be taken care of, right? So now in this particular scenario, if you see, what we exactly do is that from the mother's egg cell we take away the nucleus, and this nucleus will be inserted inside the donor egg cell. Now again, why nucleus? Because nucleus has all the individual properties of that person. So if you want the original mother's properties to be there in the child, you would need the nucleus to come from the original mother, correct? So that's why, even though mitochondria were unhealthy, we are not replacing mitochondria, we are replacing nucleus. Why? The reason is simple. we have multiple mitochondria and we have only one nucleus. So that's why if it is about replacement, replacing nucleus is easier as compared to replacing mitochondria, because otherwise there are multiple mitochondria in every cell and it will be a very cumbersome process if we have to just replace all the mitochondria. The easier process, because it's also an already established process, we know how nuclear replacement can be done. So that's why we'll take away the nucleus from the original mother, we'll replace the nucleus in the donor cell with the mother cell, and then after that the fertilization will take place. So in all these cases we see that wherever you have unhealthy mitochondria, wherever you have diseased mitochondria, we use this kind of a therapy. We use the concept of three parents baby here. Now, in the concept of three parents baby also, there have been certain controversies. So for example, whether the claim of the child should only lie with the original mother and the original father, or should the donor also have claims over the child. There was this very interesting case that happened in the UK, where it was claimed, where the child was also claimed by the donor. The donor said that because I have donated DNA to the child, I should also get the claim over the child. And that's also not wrong, because we discussed about this. If you remember, yesterday we had discussed that not only the nucleus but also mitochondria and also plastids have DNA. So that's why this person said that the donor should also have the claim of the child, because the donor also is giving mitochondria. Mitochondria also has the DNA. So the DNA of the donor also is present inside the child. The court said no. Where you are wrong here is that in case of the nuclear DNA, just remember this, the nuclear DNA is specific in nature. That all my properties, my individual properties are there inside my nuclear DNA. In the mitochondrial DNA, just remember this, that the mitochondrial DNA are species if specific, meaning that everyone who is a human would have similar mitochondrial DNA. All right. So my mitochondrial DNA and your mitochondrial DNA would be similar, but my nuclear DNA and your nuclear DNA would be different. So that's why the individual characteristics, they are coming from the nuclear DNA, not from the mitochondria. So even if mitochondria is being donated by the donor cannot have the claim of the child. This is what the court said. So that's why we see that although these are new techniques, already these kind of controversies are surfacing. So there are multiple names that we have, for example, mitochondrial manipulation techniques, or 3% in vitro fertilization, or pro-nuclear transfer technology. So pro-nuclear transfer is also another term being used. This actually was asked in your paper as well, pro-nuclear transfer. So this question was asked in your paper. In the context of recent advancements in human reproductive technology, pro-nuclear transfer is used for. I'm sure you know the answer, but still, for everybody's satisfaction, let's launch a poll. Please read the question. Please read the options. I'm just going to give you 15 seconds to answer this question. Please mark your answers. So 58% of you have marked D, which is the correct answer. Prevention of mitochondrial diseases. That is what basically we are doing. That if there is a mitochondrial disease in the mother, we don't want it to transfer to the child, and that's why we are using the concept of three parents baby. So fertilization of egg into in vitro by the donor sperm, no, that's surrogacy. That's the normal in vitro fertilization. Correct. So when we are specifically talking about mitochondrial diseases, that is where pro-nuclear transfer would be used, or where the concept of three parents baby would be useful. Then we come to another topic that has been in discussion for the last couple of years, and this is about GM mustard. Now, GM mustard, which is also called as DMH11 or Dhara mustard hybrid 11, DMH11. This is a mustard, genetically modified mustard hybrid that we have created. Now, with this mustard hybrid, our expectation is that the productivity will go up, that it will give us almost up to 25% better productivity as compared to the best of the variants of mustard that is available right now in India. And that should help us in reducing the imports, getting better productivity, help agriculture, help the Indian agricultural market. So that is the basic idea with which GM mustard was getting released. And this is something that was grown and developed by the Center for Genetic Manipulation of Crop Plants in Delhi University. At Delhi University, this is something that was developed, and it claims that it will be able to give 25 to 30% better yield as compared to the best of the variants of mustard like Varuna. So, with this purpose, the seeds were about to be released in 2018. The Genetic Engineering Appraisal Committee, they also had cleared it, and the government also then cleared it for releasing the seeds of GM mustard into the Indian markets. This is where the Supreme Court intervened, and Supreme Court put a stay on the release of these seeds and asked certain questions. That, for example, when it comes to a similar variant of DMH11, we see that in Europe it has been banned. It is in Europe, it is not allowed. So, and there are other concerns that were raised. So, because of which the Supreme Court said that no, right now it is not ready for release. We should do more comprehensive studies and research on it, and then be very sure before you are releasing it for the market. So, this is what the court said. So, then after that, we see that there was a committee that was set up. The committee started again analyzing all the aspects of DMH11. And last year, the committee uh, presented its report where it said that we have studied the things and we see that the things are fine, that things are good to go, and we can release GM mustard for the Indian markets. The government has still not taken the final decision. Right, the government has still not taken the final decision, and still is pending for the final decision whether it will be released or not. And we have to see whether or not that happens, because the concerns were a lot. For example, let's say, can it have an health implication? So whether on, let's say, short term, we understand that it might not have any implication, but on a long term, with, let's say, with 10 years of use or 15 years of use, can it have a negative impact on the health? We have to understand that better. Then what kind of effects can it have on the environment? That also needs to be understood. That, can it have a negative impact on anything else that grows along with it? Whether there is a negative impact on soil? So there are all these things, plus the biodiversity factor, that right now in India there are almost 66 variants of mustard that are being grown in India. Now, what happens is that as soon as you release GM mustard, a lot of farmers will completely move towards the GM mustard, because they will also see a better yield. So they would also want to move towards GM mustard. Then what happens? A lot of these different mustard variants, they start to become extinct because they'll not be used, and they'll slowly and steadily start to disappear. So there are all these factors, all these problems that were raised, and hence we see that it came across. There were a lot of issues that were raised there. So there was one question. This was asked in a 2019 paper. A very simple question, easy to answer question. There is one statement here that gives you the answer. So I'm launching the poll. Please mark your answers. Yes, Tikendra, you are correct. All right. So I'm just launching the poll. Please mark your answers. Okay, so 63% of you have given the correct answer. For the rest of you, I'll tell you how this question became easy. First statement is GM mustard has the genes of a soil bacterium that give the plant the property of pest resistance to a wide variety of pests. We did not talk anything about this, right? We did not say anything about this, but this is about BT cotton. This is more related to what happens in case of BT cotton. So that's why this is not related to GM mustard. Remember that in case of GM mustard, what we are saying is that this is a hybrid variety. This is a hybrid variety where different cells are being used. So has the genes that allows the plants to cross pollination and hybridization. This is correct. This is basically what we are doing. But look at the third statement. GM mustard has been developed jointly by IARI and Punjab Agricultural University. Delhi University. This is what we discussed, that the development has happened in Delhi University and not Punjab Agricultural University. So the third statement is wrong. You have three here, here, and here. So that's why you get the answer just from the third statement. We did not discuss anything about the first and the second statement while I was telling you about GM mustard, right? But you get the answer anyway. You are getting the answer anyway just from the third statement. So that's why if you read that question carefully, you'll very easily get the answer, that the answer should be B, that is 2 only. All right. Now coming to the last topic from this chapter, and this is dark biotechnology. Now, when it comes to dark biotechnology, we are talking about the negative aspects of biotechnology, the areas where biotechnology can be misused. So whether it is about bioterrorism or bio warfare, biocrimes, or even anti-crop warfare, all these things will be counted as a part of dark biotechnology. So for example, creating bio weapons or the bioterrorism part. Now in case of bioterrorism, it can be, we know that we can use biological sources, we can use biological resources to carry on attack on humans, or not only, let's say, even not in humans, let's say carrying on an attack on the crops, right? On the crops also, because let's say even if it, because bioterrorism, or let's say terrorism in general, it's not always about loss of human lives. It could be about loss of your economy. For example, let's say if agriculture, somebody attacks Indian agriculture, we know how many people are dependent on agricultural income. We know that how many people are dependent on the agricultural income, plus how much Indian economy is also developed, is dependent on Indian agriculture. So if somebody attacks the agriculture, it will be a big economic setback for India. So so that's why that also can be counted as an act of terrorism. That's why that is bioterrorism, if a biological agent is used for that. Same goes for, let's say, attacking the humans as well. So let's say if we find out certain problems with human genes, and we know that this is how we can attack humans, and this is, let's say, a virus against which the human immunity does not work, and we release those viruses in the environment, that can be dangerous. Look at what happened with COVID-19. So in case of COVID-19, this coronavirus that we had, the the immune system that we have does not have the capacity to fight this coronavirus instantly. That has led to so many loss of lives all across the world. So if at all, I'm not saying that this was produced in the lab, but there are so many claims that say that it was actually produced in a Chinese lab, and whether it was produced in a Chinese lab or not, nobody will know ever, because it's China. So you don't know what happened or what not. But there are claims, and we know that this is possible. Regenerating a pathogen or developing a virus, developing a microorganism is actually possible for us today. So that's why it's something that can be a possibility. So that's why regenerating an extinct pathogen today is something that can be done. And let's say it is used as a bioweapon. For example, consider smallpox. So smallpox, today we have an immunity, because not we don't have an immunity, we have been able to eradicate it, because there was vast vaccination that happened all across the world to eradicate smallpox. But then the subsequent generations do not have that immunity, because they haven't been vaccinated. But just that now smallpox is not found anymore, only in one or two specific cases here and there. But tomorrow, let's say somebody wants to launch an attack on humanity, Smallpox virus is one of the ways to do that. So that's why regenerating extinct pathogens is also a possibility. This is also something that can be done. Then the effects on biodiversity, that biotechnology can have a negative impact on biodiversity, something that we discussed in the context of GM mustard as well, that how we would see that GM mustard can lead to a decrease in biodiversity, because a lot of variants of mustard will go extinct because GM mustard will be used. Similarly, what we in general see is that our entire focus is only on a few species. Only a few species is where we are stuck, and others we don't care, because humans are always about plants and animals which are useful for us, right? The things that are useful. Anything that is not useful, nobody is bothered about it. So that's why we are not bothered concerned about preserving the biodiversity. We are only concerned about preserving what is useful. And that is why what happens is that only certain species like these, they will get all the care from us that's required. Others, even if they are suffering, nobody cares. So that also leads to a decrease in biodiversity. And and we see that all these can lead to a lot of problems. So, and biodiversity, when it starts to decrease, it has a huge negative impact on the ecosystems where the biodiversity belonged. So that's why it becomes a very, very difficult task for us. And decrease in biodiversity in any form is bad. So for example, why deforestation? When we talk about deforestation also, we know how harmful it can be for the environment, because you are decreasing biodiversity. And even if, let's say, for example, there are techniques, or let's say processes like afforestation, that we'll talk about, that okay, fine, we have cut one tree, but after cutting that one tree, I planted 10 more plants. So I should be happy that although I just cut one tree, but I have planted 10 more plants instead. So that's good. You have to understand that it's not, because the plant that you are sowing today is not going to grow into a tree tomorrow. It might take 5 years of time, 10 years of time, to actually get to the stage where that tree was when the tree was cut down. And what also happens is that even if we are cutting down the trees, what we plant are again useful plants, that you have cleared off a forest, and after that, uh, what you are planting are mango trees, what you are planting are neem trees, and so on. So that does not help, because you cut down something else and you are planting something else. So the biodiversity that has decreased is not really getting regained. So that is where the problem lies, that plantations are not good. You cannot say that because of the plantations, you cannot say that okay, fine, now the forest has been dekindled. You cannot. So that's why in all these cases we know that there are effects on biodiversity that this can have. And then lastly, the concerns with regards to terminator genes. Now, in case of terminator genes, what we see is that this is a kind of a gene that you insert in a plant or in a seed, that will stop further germination to happen after a generation. That, let's say, you go to the market, you buy certain seeds and you plant them. What you'll see is that one generation it will give you a wonderful growth, quality wise, quantity wise. You'll find that, that's great. So what you'll want is that you'll want to preserve these seeds for the next sowing season as well, because you have seen the quality and the quantity, so you want to repeat the same next season as well. So now in the next season, when you plant these seeds, you'll see that they will not grow, they will not germinate, because terminator gene has been used. And if terminator gene has been used, that means that further development after one generation does not happen. The seeds only germinate once. All right, the seeds only germinate once. So then in that case, you will have to then again go back to the market. You will have to go back to the market, buy the same seeds maybe, and this time the seed cost would have increased. So there are all these issues that we have with terminator genes. So that is where the problem lies. That is where we see that this becomes a big issue altogether. And not only the fact that this seed does not germinate after one generation, but it may so happen that these plants also cross pollinate with other crops that are growing in that particular field. Then the same terminator gene might also jump from one species to another, and the other plant also might not germinate in the next season. So that's why it can have cascading effect for the farmer. and the farmers would not know that there was a terminator gene, because the seed companies would never tell about this. They will not say anything about it. They will simply use it for their own business, which is unethical. But unfortunately, these are things that happen. So that's why you need to understand that these are all these issues that we have, uh, where terminator genes, we have seen that in the past also there are seed companies that uh, have been said that they actually make the use of, they have used terminator genes in uh, their seeds. So that's why understand the problem of terminator genes and how exactly they can lead to certain issues like this. All right, so this is about this particular chapter of biotechnology. We have discussed a lot of things. So again, revise everything, whatever we have discussed. If you have any doubts, you can always come back and ask those questions. Now, what I want to do next is also discuss some of the mains based questions from the previous topics that we have discussed. So, especially from space technology and energy, these are two chapters we have completed. So, just to give you a basic idea about how to handle these questions and what needs to be done when you get these questions in the examination. When it comes to science and technology as a chapter, as a subject, we see that the questions are not very difficult to answer. They are taken from areas which are fairly visible areas, either from the static areas itself or from current affairs areas also, which are very big news articles. We often see that the questions have been taken from these areas. Now, when it comes to these questions, all right, when it comes to these questions, I also want to tell you something, that more than the strategy of how to write answers and what should have been written and what you have to do, it's more about your practice. Answer writing is more about your practice than anything else, than what I am saying or any other faculty for that matter is saying. Because we can keep giving you strategies all day long, and it does not help until the time you start to pick up your pen and paper, and you start putting that pen on paper and write answers on your own. Because even if you think that I'm not prepared, I've not read this subject, I've not read this chapter, there will be a day when I'll be fully prepared and then I'll start writing the answer. That day does not come. That day never comes, and that's why you can't rely on that one day. You can never be, I'm just being very blunt, you can never be 100% prepared for this examination. Ask anyone who has been preparing for 3 4 years now. Just ask them whether they think that they are 100% prepared. Or even if, let's say, just go and ask rank one of last year or the year before that, whether they think that they qualified because they were 100% prepared. Nobody can be 100% prepared. You can be prepared for whatever you have been taught, whatever you have read, and based on that, what can you do? You have covered all the parts of syllabus, great. You have covered whatever was taught in the class, great. You have revised from the material, etc., great. So after that, you have to start. You have done current affairs, you can start. So that's why, just understand that when it comes to questions, it's not only about the strategy for these questions, but rather it's more about your practice. And this is also a point that I want to make here. This is one of the most common mistakes of any aspirant who's preparing for this examination. What happens is that students come to the class, they sit in the class, they make all the notes possible, they read everything that we are giving the class, but they never go beyond that. Beyond that is where you have to start your own preparation. Until the time I am saying something in the classroom, until the time it's my PPT and my annotations and my notes, your work has not started. Your work starts after this. Sitting in the classroom is just the beginning. Your work starts after you have gone back, after you have started revising, after you have started practicing, after you have started writing on your own. This is where your effort starts. Until then, it's all my effort, sorry. So please ensure that this is something that you start doing, because we have seen this year after year. It has been 10 years since I have been here, doing this year after year, the same kind of mistakes. Even after telling the students about a lot of these problems, doesn't help. And Many a times what happens is that there's a popular saying that you learn from your mistakes, right? People say that you learn from your mistakes and you grow and you succeed. Don't learn from your mistakes, learn from my mistakes, learn from others mistakes. That's a better approach. Why to waste your time in first doing that mistake and then after that starting to rectify that mistake? Because what happens is, in a lot of students who have been very sincere as well, we see that they clear the prelims examination, they go on the mains examination, and that is where for the first time they start writing. And this is where they realize that they should have started writing their own answers a long time back. Then the next year they try to rectify that mistake. So don't do that. Don't try to learn from your mistakes, try to learn from anybody else's mistake. That helps you better. So please start doing this, because I see this year after year that this will happen all the time, that you will simply keep on collecting all the notes, all the annotations, all the material and ARMs and whatnot, and you don't study. I mean, you don't practice. Even if you study, you revise, you read, but you don't write. When you start writing, you will identify the areas where you have the problem. And you will find that writing an answer may not be as easy as you think while reading it. For example, even if I give you a very, very simple answer, a very, very simple question to answer, still you might not be able to write an answer. For example, let's say if I ask you to write 100 words on your state, whatever state you belong to, I ask you to just write 100 words on your state or maybe on your city. Just imagine how difficult it can be if you have to write just 100 words. If I'd asked you to write an essay on your state, perhaps this was an easier question. But when I ask you to write the same thing in 100 words, that is where the problem begins. Because first and foremost, you have to write about the history of the state, the geography of the state, the politics of the state, the economy of the state, the art and culture, the demography. So there are so many things that you have to write about, and all in 100 words. How do you do that? Even if there are 4 or 5 broader topics or broader subheadings, even then writing an answer will not be an easy task. So that's why it's all about practice. Even if you know the information, even if you know what has to be written, even then writing is not that easy. So that's why writing needs a lot of practice. And I'm not saying that you have to write 5 answers in a day. There are weekly tests happening. Please write those answers. Even if you can't do that, pick only 2 questions out of it, write an answer on it on a weekly basis. I'm not saying even daily basis, but it has to be done. And please understand this as early in your preparation as you can, that it's not about the strategy that I'm going to give you in writing your answers. It's about your practice. It's whether you are doing or not, you are doing a sincere effort, you are making a sincere effort towards writing an answer or not. That is what will really help you. Otherwise nothing else is going to help you. No strategy is going to help you. As long as you have a good content and you can put that content on the paper, that's it. It's not that you have to write wonderful answers. Just go. Have you seen the topper copies? I mean, there are so many. You simply go on Google and you just look for the answers of the previous year toppers. Have you seen it? Yes, many of you are saying that you have seen it, right? So I have a follow-up question. Do you think that that was the best answer that you could have written? After studying something, you might many a times feel that it's not the best answer. You'll just think that, okay, fine. uh, Sometimes you might even feel that how is it possible even that this person got rank 1 after writing such answers? So that is where you have to start understanding the beauty of writing a good average answer instead of trying to write the perfect answer. Because if you start let's say chasing a perfect answer, you might write some very good answers, but you will never complete your paper. That also is something that matters, that whether can you write answers to all the 20 questions that have been given. More often than not, when you go to your mains examination, you will be very, very well prepared, because you have a lot at stake. You'll be very well prepared. After prelims examination, between prelims and mains, you have that time where you will be preparing very well, ensuring that you have covered everything possible. So that's why there are chances that you know the answer to all the 20 questions there. So now whether you are able to complete those 20 questions with every piece of content and every point that you wanted to mention in that answer is what also matters. So all these things need a lot of practice. That's why I'm focusing and begging probably in a manner that you have to keep practicing. Nothing else works as much as your practice works. So that's why this is something that you need to do. You have to start writing, you have to start taking that difficult step, taking that uncomfortable step where you are writing the answer, and submit those answers. Submit those answers on the email IDs that we have provided you, so that you can get the feedbacks on these answers as well. That's also very necessary. And write a horrible answer, nobody cares. Nobody is going to chase you down and come to your home and say that, what is this, why did you write such an answer? Nobody is going to do that. But write an answer, start writing an answer. Be embarrassed about that, writing that answer, that's fine. Nobody is going to question you, nobody is going to say anything. It's not that we are going to publish that answer on our website and say that, look at this horrible answer. We are not going to do that, right? So it's all about your personal growth. While you write, start writing a bad answer, and from there on, how do you get to an average answer, and then an above average answer, and maybe an excellent answer towards the end. But it takes time, it's a journey that you take. Maybe some of you are naturally gifted and you start writing good answers from day one. Great if you can do that, but I know that many of us can't do that. So we need that journey. We need to start from zero, or whatever answers, whatever horrible answers we write, and from there on start developing, understand, based on that feedback, based on whatever is being told to you. Maybe if, let's say, the feedback that has been given, you don't understand that feedback, call up the mentors, say, tell them that, okay, this is the feedback given to me, I don't understand this feedback, tell me what exactly should be done. So all these things is for your own personal growth and nothing else. So that's why you have to do this. I mean, that's why I'm saying, I mean, uh, that more than what I'm going to tell you here, it really matters whether you do it or not. It's about your effort, it's about whether you are making that effort to write an answer or not. That's the only thing that matters here, and nothing else matters. So anyway, let's look at some of these questions that have come in the past papers. So if you see the first question, now when it comes to space technology, you'll find that almost every year question does come from space technology. There are only certain years which have been exceptions. For example, the COVID years 2020 and 2021, questions from space technology were not asked. COVID related questions were asked. So we see that there are certain things that basically uh, certain areas where space technology have not been asked. Otherwise, most of the other years we have seen that space technology questions have come. 2022, launched on 25th December 2021, James Webb Space Telescope has been much in the news since then. What are the unique features which make it superior to its predecessor space telescopes? What are the key goals of this mission, and what are the potential benefits that it holds for the human race? So it was a straightforward question and an expected question. We actually expected that a question would come in the prelims examination. In prelims, when they did not ask this question, we were expecting that a question would come in the mains, and that is what happened. A simple direct question based on the properties of James Webb Space Telescope. For example, it is an infrared telescope. It has very highly sensitive mirrors and lenses. It will be able to give us an insight into the early galaxy because the waves can travel very long. It has been placed in L2, so that provides it an extra shielding from the harmful radiations of the sun. So with regards to the predecessor, so predecessor was, for example, the Hubble Space Telescope, or even if you can name the Spitzer telescope, which was the previous infrared telescope. So in all these cases, we see that compared to them, there are a lot of qualities that is superior here. At the same time, it will be able to give us data and knowledge about the things that, you can say in a manner, have been unknown to us all this while. So there are a lot of advantages to draw from. So a simple question, it was related to current affairs of 2021-22, and expectedly a question came from there. All right. But just remember that there are three parts to this question. First is about the unique features. How is it superior from other telescopes? What are the key goals of the mission? And what are the potential benefits that it has? So there are three parts to this question. What we will do simply is that we start with a simple introduction. We start with a simple introduction into the launch, that the launch was done and it's a big launch. And then after that, three subheadings. that what are the unique features, what are the key goals, what are the potential benefits. So in this manner, you will take three subheadings and then conclude and talk about, let's say, a potential future. So it's a simple question where you can very easily write the answer, and it was related to that year's current affairs. Then in 2019, what is India's plan to have its own space station, and how will it benefit our space program? So again, two parts here you can see. One, what is India's plan for own space station, and how will it benefit our space program, right? Benefit our space program. So this was again based on current affairs, because in 2018 there were speculations that India might join International Space Station. India might become a part of International Space Station. This is where India clarified that we are not going to become a part of International Space Station, but rather we are working on our own space station. And by 2030, we are targeting that we should be able to have our own space station ready. So this is basically the context in which this news basically was, this news had come. And it was announced that after we are done with Gaganyaan mission, after Gaganyaan mission, the next big thing that we'll be working on would be our own presence in the space in terms of our own space station. Now, what are the benefits? Benefits are multifold. For example, we'll be able to carry on a lot of experiments, understanding of the space environment, the effect of space environment on animals, plants, and humans, how our biology responds to it, what is microgravity, and how does microgravity uh, work in space, what are the things that we can do to counter that microgravity so that our future space missions can be helped. Similarly, to provide a support to all the low Earth orbit satellites. The low Earth orbit satellites, the polar satellites that we have, we can provide a support to all these satellites as well with the help of this uh, space station. So there are multiple such things that we can do. Then after that, another simple question that has been asked is based on IRNSS. That why is IRNSS needed? We discussed about this, and how does it help in navigation? So how exactly the navigation happens with the help of seven satellites, the triangulation process, the standard positioning service and the restricted service. So you can mention all these things, the kind of security that it provides, the internal security aspects, plus how is it important for us in defense and regular positioning services as well. So there are a lot of things like this that we can write in such a question. So again, an easy question, direct question. Next question, if you see, India has achieved remarkable successes in unmanned space missions, including the Chandrayaan and the Mars orbiter mission, but has not ventured into manned space missions, both in terms of technology and logistics. Explain critically. So now this is basically, you can say that in a manner, this is in the context of the Gaganyaan mission, because Gaganyaan mission was already uh, in in the making. The announcement was about to happen, but Gaganyaan mission was already in the making. We see that there were multiple experiments that were conducted with regards to the Gaganyaan mission, and this is the context in which this particular question was asked. Now, when it comes to the Gaganyaan mission, what we see is that there are a lot of technical challenges that we have encountered, and we have been trying to solve all these issues. So technologically, we need to, until now we haven't used anything of this sort, but other countries have. So we learned from Russia, we learned from other countries. At the same time, logistically also, cost wise also, it's a big project. The total, for example, the total budget of this project is approximately 10,000 crores. So that's why logistically, cost wise, all these things also we have to take care of. Plus technologically, a lot of things. For example, you have to ensure to bring back the astronauts who are being sent. So that technology until now has never been used, so we are testing those technologies. At the same time, we want to spend some time in space. How exactly do we do that? What are the features that the space suit should have, or the helmet should have, or what kind of nutrition is required, each and everything. So that's why there are all these challenges that we have to look into. Then look at this question: discuss India's achievements in the field of space science and technology. How the application of this technology has helped India in its socio-economic development. Again, a very simple question. because we are talking about all the achievements. So there are two ways to write the answer to this. The achievements could be historical, right? You can historically keep on saying that this happened, that happened, this happened, that happened. So you can keep on mentioning all that, that our own satellites, we had our own launch vehicles, PSLVs, GSLVs, cryogenics, communication satellites, IRNSS satellites, so many earth observation satellites, Mars mission, Chandrayaan mission, Astrosat mission. So there are so many things that you can write. All these are achievements, right? And then how has it helped in socio-economic development? So socio-economic development, how has it helped agriculture? So better agriculture, better produce, better productivity, better knowledge of what to grow and what not to grow, better communication available now, telemedicine, tele-education available, IRNSS also helping in defense and navigation services. So more jobs being created, more employment opportunities being created, more revenue being generated by India, more startups coming in this field. So multiple such points that you have, that you can write. And this was asked for 200 words, because up to 2016 all the 20 questions used to be of 200 words and 12.5 marks each. After that we have seen a change, that there are 10 questions of 10 marks and there are 10 questions of 15 marks, the current way in which they are asking the questions. And then lastly, another question that has been asked is, what is the meaning of standard positioning system and precision positioning system in the GPS era? The advantages that India perceives from its, again, the same IRNSS program. So again, the same answer, the same question, and we have discussed this, that standard positioning service and precision positioning service, which is also called as restricted service. So we had discussed all these things in the class as well. So a simple direct question has been asked on it. So space technology, that's why remember that almost every year we see that questions do come on space technology. Then there are 3 4 questions that have come on energy, and energy especially, you will see that more questions have come in the past few years than what used to come. So for example, 2022, do you think India will meet 50% of its energy needs from renewable energy by 2030? Justify your answer. How will the shift of subsidies from fossil fuels to renewables help achieve the above objective? Now, whenever you get such a question where they have asked, do you think India will meet 50% of the targets? You have to basically present objective points. It is not a yes and no answer. No, definitely not. You cannot say no. India can't. You can't say that, right? In a answer, you can never write that. But even in case of this question, it's not even a yes answer. You cannot say that yes, India will achieve without presenting your points. So you will have to give your points that 2022, this is where we are. 2030, this is what the target is. These are the schemes that are being run right now. With this, we are trying to achieve this. We are trying to achieve that. Green hydrogen, nuclear energy, solar energy, wind energy, all these things, they all have to be taken into account, and you have to write a balanced answer. And you can say that, okay, fine, these are the things that we are doing, and if we are able to do this thing, or in a similar manner how we are going now, uh, there is no reason to suspect why we can't get to 500 gigawatt of our target, or 50% of the energy needs to be targeted by 2030. So there are all these points that you have to objectively present. And then shift of subsidies. Will shift of subsidies help? Yes, of course it will help, because when you shift the subsidies, you are basically providing extra financial support to that system. So that extra financial support will always help. It will always help. Not only the fact that you are boosting that budget, but at the same time we'll see that a lot of private firms will also start moving here, right? The private firms will also start moving in this direction, because they also find that benefit. They also know that they can actually get benefit out of it. Then Nobel Prize in Physics 2014, and this is about the blue LED. And how has this invention impacted the everyday life of human beings? Now, blue LEDs are everywhere, whether it is lighting of your homes, whether it is your electronic screens on which you are watching the lectures, whether it is in the medicine, whether it is in the healthcare sector. We see that everywhere blue LEDs today are being used. And blue LEDs, over the period of time, they also have become cheaper. Today we also see that blue LEDs, they the blue LEDs also uh, the lifespan, for example, of the blue LEDs are also better compared to the incandescent lights. The electricity bills that you get is also lesser, so it also saves you a lot of energy, and there are all these advantages that we have. And hence we have seen that all of a sudden so much of use is happening. Here you can also name the schemes, for example, what the government of India has. So let's say something like an Ujala scheme can be named, that how with the help of Ujala scheme in the rural areas especially, LEDs were provided for free, so that we can change the entire setup, we can change the entire ecosystem of the incandescent lights. So that's why this question. And then these two questions, if you see, these are two questions which have been asked on energy, right? And both of them are based on nuclear energy. That first says, with growing energy needs, should India keep on expanding its nuclear energy program? The answer cannot be no. You cannot say that our energy needs are growing, but we don't need nuclear energy. But it talks about the facts and fears, the positives and the negatives. What are the facts? What are the positives? And what are the issues that we need to consider and we need to overcome if we want a good growth in nuclear energy? So, again, we discussed all these points. Give an account of growth and development of nuclear science and technology in India. What is the advantage of fast breeder reactor program in India? So we discussed about the three-stage nuclear program. That precisely is what we have to mention in this particular question. So again, a direct question on nuclear energy. All right. So these are certain questions that have been asked. Please practice. Please write the answers on your own. Write the answers, submit to us, get your own understanding of where you are and what you are doing. That's more important than anything else. All right. So please do that. So next, I mean, in the next class tomorrow, we'll start with nanotechnology, artificial intelligence, and robotics. These are the things that we are going to discuss in the next class. If you have any questions, please let me know. Let's discuss them, and then we'll close the class for today. This is the leaderboard for the day. You can have a look. What is a tail word? Examples: explain, critically, elaborate, illustrate. Yes. So see, each and every question or each and every tail word has its own way in which you have to answer the questions. So for example, when something is like describe or explain, so then in that case you simply have to give objective points without putting any kind of opinion, positive or negative. But when it says examine or critically analyze, so in these kind of cases what happens is that they are also probably asking for your own opinion, but that opinion also cannot be your own opinion. Rather, it should be based on somebody who is an expert of that field and how they have expressed their opinion on something. So based on that, you can always quote people there. You can always say that this happened, that happened, and these are the facts and these are the reports, etc. So in that case, you always have to ensure that your opinion does not seem to be your own opinion, but rather it looks to be the opinion of somebody else, and you are using that opinion to be presented in that answer. The question on 2014 Nobel Prize was asked in 2021. Was it based on any current affairs topic due to which it was asked? Not really. This was also a kind of a surprising question, but because blue LEDs now have become part and parcel of our lives, so that's why I think that question has been asked in a manner, you can say, the advancements of technology that has been happening. But I wouldn't call it as a current affairs question because it was a 2014 current affairs question. How to scan an answer for sending on email? I think there are scanning apps, etc. They are available, or I think the cameras can also do that job, and that you can collate into one single document and you can send it. Difference between hybrid and GM crops. So hybrid can also be uh, counted as GM, but in case of hybrid, what we are doing is we are just trying to mix the genes. We are more trying to mix the genes, and through cross pollination, etc., we are just trying to mix the genes between species. But then there are genetic editing that we can do. So that's more specific towards GM crops, where we are modifying the genes artificially with the help of recombinant DNA technology, etc. Prakash Path scheme related to blue LED. Yes, you can say that. Good handwriting helps. I would say the handwriting helps only until the point that your handwriting is readable. It should not be that I can't read your handwriting. Having very good handwriting does not really have any positive or negative impact. Just that your handwriting has to be readable as much as possible. That's it. Should we read biotech's NCRT of class 12th? No, not required. Are sterile seeds a product of Terminator genes technology? Yes. In most of the cases, yes. How is therapeutic cloning done? We discussed the same somatic cell nuclear transfer technology, just that at the end, after the blastocyst is produced, you just take that blastocyst and you store it. Had sent my answer, didn't get feedback due to some guidelines. Please check with the team why exactly that happened. What are VOCs, volatile organic compounds? See, volatile organic compounds are also counted as air pollutants. A lot of organic molecules, volatile meaning that something that can boil very quickly at lower temperature. So in these kind of VOCs also we see that they have a big impact on the environment, especially on the air, and that's why they are a concern for us. Apart from NCRTs, which book prefer should we prefer for science and technology? None of the above. No other book is required, because otherwise it's very distributed. The subject is very distributed. A book does not help there. Once again, happy Teachers Day. Thank you so much for everyone who has wished Teachers Day today. Means a lot. The issues with regards to books that some of you have, please write to the support team. They might be able to help you. Yes, referring to class and slides are enough when it comes to in most of these cases. All right. So I think that's enough for today. Please go back, revise everything that we discussed. Please start writing. Please start writing answers. Please give it a try. For those of you who are doing it regularly, wonderful. For those of you who are still waiting for that one fine day when you think that you'll be 110% prepared for this exam to write the paper, or you are afraid that what happens when somebody finds out that I'm not writing a good answer, nobody is going to do anything. We are just going to provide you a feedback, and that is only going to help you in your future. So please start writing. Please start writing, and it is only going to help you and nobody else. So please do that. So with that, let's end the session for today. Please go back, revise. If you have any doubts, please bring them back, and we shall discuss that tomorrow. Thank you.